ఇంకొకడు శాలరీ రాలేదట లాస్ట్ మంత్ అంతకుముందు చేయకుండా పని చేస్తుంది రాలేదట. హలో హలో
హలో హలో హలో హలో శ్రీమన్నారాయణ. హలో 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 శ్రీమన్నారాయణ శ్రీమన్నారాయణ శ్రీమన్నారాయణ
नया वाला पुराना वाला खत्म हो गया। तो चेक किया वो कौन सा है? छोटा वाला भी है 5 kDa का, 8 kDa का, कौन सा निकाला? अगर 5 kDa 8 के 5 तो किसी और के लिए है ना, ये पीपल नहीं लिखा होता है ना।
morning.
Yeah, good morning, everybody. This is Sai Kumar from IVRI here. I just logged in.
I see you.
Yeah.
Looking good.
Oh, yeah. So you are sitting in a group, I guess, NIAB. Yeah. Good morning, sir. Good morning, Director, sir. Dr. Triveni, sir.
Morning, sir.
Very good morning.
Hi, morning, Bhaiya, Dr. Triveni.
Good morning, sir. Good morning, sir.
Nice. In such a big auditorium, only 3-4 people. Gujarwala sir, Namaskar. Madam, and also to Madam here there. Also fine here. Good to see you all once
Not
again.
your madam. Yeah. NIV madam.
NIAB madam. Yeah.
Triveni ji ठीक है?
Yes.
हाँ.
Triveni ji बढ़िया. ठीक है. Dr. Triveni, everything is fine?
Sir, everything is fine, sir. Good to see you, sir. in a full energetic mood.
I specifically came to see Dr. Taro.
Good, sir.
Hello, Dr. Balamurgan. Good morning.
Good morning, madam. Good morning, madam. How are you? Fine, fine. Thank you.
Some of our people join on. Hare Krishna, AC is not.
एसी चल नहीं रहा भाई, वो मैं देख रहा हूँ इलेक्ट्रिक का कोई आदमी नहीं। सर जब भी आते हैं एसी का प्रॉब्लम क्यों होता है? पहली बार तो हमारी एसी था ही नहीं। They have put it on just now. It will take time. Actually, nobody sound that well. So why our people have not come to auditor?
Some of them have joined from the labs, I can see.
Hey, Deepali Rawat, why?
Dr. Anand Srivastava.
Good morning, ma'am.
Good morning, Dr. Anand. We are waiting for you in auditorium, Dr. Anand.
Okay, I'll come there.
Also, students को भी थोड़ा बोल दीजिए.
Please tell your group students as well.
Sure, sure.
Our students are also joining us.
Oh, they have joined.
Online है. Priya Gupta है, Deepali Rawat है.
कौन बोल रहा है? डायरेक्टर सर ना?
इसी का
प्रिया गुप्ता.
Now, if it turns on, it's on good. Vishnu, call Ramesh Prabhu immediately.
रिटर्न प्लान आती है उसमें आज ही डायरेक्ट फ्लाइट नहीं वहाँ पे फ्लाइट डायरेक्ट फ्लाइट इज इन द मॉर्निंग early 9:30. If I go today, I will really go early tomorrow. So no use. What airport waiting for you? It will go. Flight is there. First, it will go to Chennai. Then it will go to Delhi. Okay. Then it will go to somewhere. Then next day morning to Guwahati.
কালি স্যার ডক্টর বর্ণমানটো মিটিং দিছে আমাৰ এই প্ৰজেক্টৰ মই ইন পাৰ্সন যাব নোৱাৰিম কিন্তু মই আজি ৰাতি 2টা বজাতে ফ্লাই কৰিম স্যাৰ
করলে
ডিব্ৰুগড়লৈ
রাতে
3:55 ত সৰি এটা ফ্লাইট মৰ্নিং এই কলকাতা আৰু 1 ঘণ্টাৰ পিছত ডিফাৰেণ্ট ফ্লাইট বাট 10 o'clock I'll be there morning Indian Airlines said it অলপ আৰাম হৈছে
So you are going to Guwahati or Dibrugad?
Dibrugad jam, Dibrugad at least online. More as a co-investigator of physically. So you will be there.
What it is? Going at 10:30, okay, 3:30 in the morning is very good. You could have
disturb the whole.
You could have gone in the morning flight, 9:30. The flight I am taking. That will reach 11:30.
So they're having this medical thing in the first half.
Well, that is would have planned. 11:30 means by 12:30 you would have been in Kanapa. Okay. Even then that is much better.
Cost over.
Yeah, the whole night.
ग्रुप को टिक कर दो जिसमें एसी लाइट चाय तो वो सब उसमें सब आ जाएगा तो क्या है कि एक क्लिक में सबको पता है क्या करना है
So, very good morning to one and all, and I heartily welcome Major Barua sir and Siraj Ahmed sir who took time out to come over here, and he is physically present for this meeting. And I also welcome our director, Madam Sujay Majumdar sir, Hegde sir, and other faculty members, students, staff for joining this program in the morning. I also welcome lots of distinguished scientists, panelists, those who have joined through virtual mode in this morning. So this is a very important workshop that we have organized, that is on genetic diseases with special reference to leptospirosis and Japanese encephalitis, where we want to discuss about the current status, challenges, and the way forward. So it will be really great to hear from all the distinguished scientists and panelists to get some ideas that how we have to take it further. So to begin with this program, first I would request Director Madam to please welcome the guest, Dr. Bejar Valu, Chief Guest, Bejar Valu. Now to begin with the program, may I request Dr. Sujay Majumdar sir to please to talk about the introduction about the flagship program that we have here.
Good morning and welcome to our respected Major Barua sir, who is not only great well-wisher, but he was SAC chairman for all the progress we have done until now. He is under his direction and suggestion and guidance. So he is part of the accomplishment of NIB, legally, officially. That's why with any call he comes, and we are grateful to him. We are grateful to Dr. Siraj Ahmed, who is a happy-go-lucky guy, like one of us, and has taken the train to come here. And let me mention that tonight, probably at 12 o'clock, he will go to the airport and stay there up to 3 a.m. and catch a flight to go to Kolkata. From there, he will get it. But still, he has come. So thankful to you. So this particular program, it was given by Department of Biotechnology. They asked us to have a flagship program. Since we already had a flagship program technically of the lifestyle genomics, so we played a clever role. We had hidden lifestyle genomics, and suddenly we had a flagship program, which is pathogenomics. So the word came in when Ravi, myself, actually first did the mind-boggling thing, what to write, and then we included around 5 scientists and 5 diseases in that. And we went to fight to DBT. DBT turned it down. And when turned it down, luckily we were there. I was also sitting, and my name was there with OPI. So then we argued with the chairpersons. And what I argued is what this project is. So they said the title is wrong. I said yes. Then they said, so then inside things will be also wrong. I said yes, obviously yes. You rejected in front of me. And Dr. S.R. Rao was there. Even before starting of our presentation, he said there are people who are trying to become Nivedi, another whole Nivedi, and giving us a program. So I understood he is telling me. But just before presentation, he called me, Dr. Rao was the program officer of DBT. And when I went, then he said, "So we क्या project लाया है? सब कुछ तुम तो कर लो, जो नहीं भी करता है, सब समझ जाना आपका है." So the chairman rejected the proposal as probably pre-decided in my view, whatever. But after rejection of the proposal, I argued that, sir, proposal is not what you are thinking. Proposal is what I can say. Then I said, but why title is wrong and why you accept that inside it is wrong? I said, on Friday, it says, on Monday, you submit a 5 crore grant. This is going to happen. So understand what I'm trying to say. He said, okay, go ahead and say. So luckily, those chairmen very closely knew me. They both were from Delhi and they have watched me and they knew that I have substance and they, after rejecting project, they listened to me. Then I said, the whole program is about whenever we go for host and pathogen interaction, there has to be a tissue, whether it is a blood cell or a kidney or lungs in case of leptospira or in case of JV, for example, brain. And we went with 5 diseases, as I said, brucellosis, tuberculosis, leptospirosis, JV, and I think the toxoplasma gondii. And then they said, I said, what do you want to do? I said, when the pathogen attacks the host, the area of the host where pathogen is actually going and sticking and attacking, the whole transcriptome, the gene expression, everything changes. Some of the molecules are facilitated of the pathogen to come in and come in. Some of them are restricting pathogen to not come in. Same pathogen also interacting with the host. We want to pick up those genes by either differential display or DNA microarray or RNA-seq or whatever. Then the major problem comes, there are 2,994 genes upregulated, 3,224 genes downregulated. What do we do with that large number of genes? So I have had one paper at that time in DNA research. One of my students was very brilliant. I'm not expert of this field, but then he taught me a lot of things, that how you can do various kinds of proteomics and genomics and how there are programs which can bring them all from 3,000 to say 12 genes. And for example, if there are 110 genes which are regulated by one transcription factor. So rather than studying 110 gene expression up or down, you study one transcription factor. And if you get, for example, for 3,300 genes, 36 transcription factors which regulate them. So you have come from 3,300 to 36 transcription factors. Now, this Kamal Mandal, very smart guy, he said, sir, 36 को reduce करना है. हम लोग तो layman थे. So how to reduce? He said, sir, let me do cytoplasmic and nuclear presence of this. So, if out of 36, only 21 are present in the nucleus, that we should study. Because those which are in cytoplasm, those transcription factors are used. So like that, these are very simple things. This is not what we plan to do in this thing. But we thought that we'll involve foreigners, and there are very nice platforms and software which we use and learn and make a platform. Like any atta chakki, you go there, put any kind of grain, whether it is bajra, gemu, jawari, whatever. Its job is to do something and give you as a powder on the other side. So if we make such platform of 4 or 5 series of bioinformatics platform and softwares and all, with little bit molecular biology, probably we can get authentically reduce from 3,300 to 12 or 13 molecules with which we can work to stop the post-pathogen interaction. That is the project. So after listening to this, they agreed. They said this is a wonderful idea, but your project does not reflect that. So we agree that we will grant this project. But why have you written 5 microbes? I said, sir, I thought we don't know which one will be lucky for us. They said, no, you do it for 2. So that way, DBT people were happy. That project is bad, it is rejected. And within 20 minutes, we created a new project which was granted, which the chairman really wanted to grant. So this is the project, and I am happy that Ravi and Sarwar did whatever they could. We tried to contact international people, 14 people, 15, because of COVID situation was different. But there was Dr. Prabhakar from Singapore, I think, who really worked a lot with us, but in between he also lost interest and we couldn't give what he wanted, so much of medical information of the patients, because otherwise he cannot make a formula or whatever. So in between that is interrupted, but the project is now extended for 6 months, but I'm sure it will get extended for 1 more year. And now Dr. Taru Sharma is handling this, and I wish the team very good luck. And I thank all the esteemed members who have joined online, Dr. Zivanita, Dr. Kumar and others. And I'm very sure that there will be a scintillating interaction. Thank you.
Thank you, sir, for giving a deep insight view of this project, how it came, and your good wishes will always help us to move further. So may I request now Dr. Hegde sir for introducing us about different kind of Gnosis that we face.
Respected Guru sir, Director Madam, Sudhir sir, Dr. Bhavute, Dr. Siraj Khan, other dignitaries online, Director IVRI, JVR, IVRI, Dr. Dinkar Raj, other colleagues, Navy faculty, students, research fellows. When I was asked to talk about zoonosis, I was actually scratching my head. A couple of reasons. One is that we have gone through so many waves of COVID now. We are pretty much fatigued. And most of us who work on infectious diseases have been invited to speak on zoonosis at some place or the other. So we are also actually fatigued to search for topics as to what to speak about. So when I was scratching my head, I came upon this paper which talks about ecology of zoonosis. So rather than talking about different kinds of zoonosis, I talk about ecology of zoonosis. Granted, some of this stuff is already there and known to people, but I thought I'd highlight this. So this was published in Lancet in 2012, 10 years ago, talking about ecology of zoonosis. So what are the different aspects of disease dynamics? We have to first understand that pandemics are rare. We don't come across pandemics that frequently. The last one we had was in 2009, and then we had in 2019. Of course, it's more frequent now than before, but they are not that common. But zoonoses are very common. They happen very frequently in different places. Many are actually enzootic in animals. And in many cases, there is no person-to-person transmission. For example, rabies, trypanosomiasis, and one of the diseases started today, that is Japanese encephalitis. And only some become serious outbreaks. For example, Ebola that happened 6 or 8 years ago, and global problems, including pandemic influenza, COVID. And endemic diseases in one time or area could be emerging in another. One example, in terms of time, organisms can take new traits, and one famous thing that we can talk about is AMR. So the same organism could, gain drug resistance for whatever reason, and so that in an earlier time they were not problematic, but now they are. Or some organisms may cross species. SARS-CoV-2 is probably a good example, or geography in some cases. Nipah is a good example. And today's sporadic could be tomorrow's pandemic. So everything starts with a sporadic disease. And then when the transmission is high, it becomes a pandemic. And diseases that are local today could be global tomorrow. And there are several reasons for it. So what is the abridged ecology of zoonosis? I'm not going to talk in detail. I'm going to point out a few things only. Zoonosis typically involves at least one pathogen, has to be there because it's a zoonotic disease, and two host species. Obviously, because it's zoonosis, it has to come from animals to humans, or if it's reverse zoonosis, from humans to animals. But it gets complicated when one introduces vectors, for example, mosquitoes, ticks, etc., intermediate hosts, and this includes things like invertebrates that spread parasitic diseases, the environment which influences, for example, EMR, and also food production practices. Some examples are given. When rinderpest was wiped out, this is the only animal disease that's been eradicated from the world, much of the cattle and the wildebeest were wiped out by rinderpest in the beginning. The tsetse flies, which were feeding on these animals in Africa, actually transferred to feeding on humans. And they also were transferring a human disease called sleeping sickness. So there was a spike in sleeping sickness simply because rinderpest was wiping out the cattle and wildebeest. So this is a natural effect. Other natural effects, El Nino, you may have heard about El Nino and La Nina, which are weather patterns in the oceans, or temperature patterns in the oceans, influence the increase of hantavirus pulmonary syndrome, mainly because there was more rain, followed by increased vegetation, because of which there was a spike in the rodent population, which actually transmits the disease. Remember that the rodents themselves do not die because of hantavirus. So there was no control or change in the population of the rodents, at least for these seasons, until the rains came down. The changes in diet in the livestock production systems, for example, beefing them up with artificial diet or things like that, could change or competitively exclude certain organisms and cause changes in microbiota and indirectly influencing AMR. And of course, foodborne diseases, more often neglected area of zoonotic diseases, are involved in transmission to humans. For example, parasites like Echinococcus, cysticercosis, bacteria such as Brucella, tuberculosis, salmonellosis. These are important pathogens that are also spread by the methods that we use for food production and storage. Of course, everybody talks about change in land use, exploitation of natural resources, including mining, logging, plantations to increase production, oil and gas extraction. All these influence human-animal interaction. And therefore, there is increased exposure of humans to pathogens. So what are the drivers of zoonosis? Of course, one is the pathogen ecology. How does a pathogen adapt to different environments, and how does it evolve within a primary host or a secondary host? However, usually the pathogen mechanisms, they rarely change, except, these changes are usually inherent properties, except when they are presented with an explosion. That's when these characteristics are also amplified. So a pathogen, we think, could change its patterns or mechanisms, but really we are looking at an expansion of the properties of the pathogen. And of course, the major reasons of anthropogenic causes, which cause large changes and providing faster opportunity, as I said, for pathogen adaptation. This could be population and recreation. This could be socioeconomic and behavioral structure of human beings. It could simply be land use, extraction of natural resources. It could be food production systems, simply travel and trade, or even use of vaccines and antimicrobials. One example here is the Streptococcus pneumoniae in humans. We know that initially the antimicrobial resistant strains were reduced, but then the other strains have started coming up. And of course, antimicrobial resulting in AMR. And of course, in the third world countries, food health systems to detect and contain these pathogens. So I'll just put up two figures. The first figure is here, where if you look at the spillover events are frequent and common, but they die off. Sometimes you have a spillover from wild animals to domestic animals, where there is amplification of the pathogen. And during this time, again, sometimes there's a spillover with human beings and transmission within the human population where you see human disease. So basically, what we get from animals is very small compared to what frequently happens in wild animals. So how to address zoonosis? Number one is there's a need to understand the evolution of pathogens, which is what this workshop is a little bit about. And we need to actually manage human-animal interface. We cannot get rid of it. We cannot avoid human-animal interface. We depend too much on humans for our food and other purposes. We have to change our behavior and socioeconomics around it. And there's a need for control of zoonosis through surveillance, forecasting or forewarning, and early detection. And these are actually the key to controlling zoonosis. And for which we really need intersectoral collaboration. And this is easier said than done. So what we might achieve, so these spikes will remain. Of course, the transmission to livestock or domesticated animals will also remain. But if we actually institute forecasting or readiness, or even early detection and control measures, including rapid response, we might actually reduce this spike in humans. So this gray area here is actually the benefit for the human population. That would be the consequence of control or study of zoonosis. With that, I'll stop. Thank you.
Thank you, sir, for such a nice illustration about the Gnosis. Yeah, and we have another dimension to look at from that perspective, also the way you have presented. So now moving ahead, I would like to request our director, Madam Dr. G. Taru Sharma, to brief about the institute and the research activities.
Thank you very much, Dr. Pankaj. Good morning, greetings from now, Hyderabad to everybody. Most respected Dr. Vijay Barasa, the chief guest of this morning's workshop. The guest of honor, Director IRI, Dr. Kiweni Dutt, having accepted and who's already joined with us. Dr. Sudhir Majumdar, Distinguished Professor NIA, Dr. Bhargav, Director and RC in Hyderabad. Dr. Siraj Khan, who has joined us for this workshop. My colleague, Dr. Hegde, Head of Academic and Research. Joint Director Research, IDRI, Dr. Dinakar Raj, Dr. Balamurugan, and all people who have joined us online and who are present out here in the hall or in the laboratories and who have joined us. A little bit, very quickly, what NIA does and what exactly the vision and mission of NIA. It's a very happy morning that our former SAC chairman, as Dr. Sudhir has told, under whose guidance the institute grew in various dimensions and research areas. And we also have today one of the current SAC members, Dr. Dinakar, with us. We really value your presence, so both. This is a journey, it was stamped on 25th May 2011. So we are yet to become 11 years old in another 2 months' time. And that's how it was started in a small area block. And this was near 4 days. And this is what we are now. It's a Republic Day picture for all of us. These are our key areas of research. We work in reproduction, infectious diseases. This is a group, and all the diseases which are pointed out over here. We have a group which is rich in reproduction and to address infertility. And then there is a group which works on, and Dr. Majumdar's laboratory is known in the area of transgenic biology work. Then various therapeutic molecules, and then we started very recently, last 2 years or so, this institute on stem cells, and when I joined, I also joined hand with the team. We have a very, very good group of bioinformatics colleagues out here, and so it's for the nanotechnology and nutrition and nutrient nano delivery. We are coming up, and there are upcoming projects which we have. As I said, that we work in this area where infectious diseases, different bacterial and viral diseases, besides reproduction, nano delivery, nanotechnology, biosensors, animal biofarming, as well as the bioinformatics. So in a nutshell, I'm not going to read all that. So what exactly we achieved in 21-22? Livestock genomics remains our pride, and more than that, the IndiGo, which is India's first genomic, first cattle genomic chip, and the world's largest chip, having more than 788,000 sequences there. And this was released, so many people know, but some of the ones who have joined for the first time to our institute activity, I like to highlight. This was inaugurated and it was unveiled by Minister of Science and Technology just a day before Independence Day. And the institute does have a very important ongoing activity to One Health Consortia, besides other important outcomes which we have. We have very many leads in an area of diagnostics, and one is looking forward for generating a very crisply vaccine candidates, besides working on a production aspect, especially our reproduction biotechnology group was robust, but then off late the emphasis was given on a nutrition biotechnology, nutrigenomics to be particular. And then good thing happened when we, one of the colleagues joined who has an expertise in nanodelivery, and we generated some important programs, especially, Riemann bypass if we had to reach the protein for the viability of the animals, likewise. And this is our focus for 22-23 by now. I think you must have read what I wrote in capsule fashion. This is all our focus is, and we are moving ahead in a very focused way with our Mendelian objectives. So to just share some of the major outcomes and ongoing research over here, quickly into different, this very workshop, which is on pathogenomics of two important pathogens of leptospira as well as Japanese encephalitis. And that's our flagship program as was detailed by Dr. Shadi. Besides that, we have the work going on into this area, and these are the leads which I showed to you. Besides, we have a very laudable outcome from for some of the diagnostics, out of which some we have already gone in a talk with the industry, and some we are about to do that. And maybe in 1 month or 1.5 months, we shall be doing so. So just to talk about the One Health Consortia, which is there, as Dr. Hegde was pointing out, that more important is to be at it. So we are very much at it, and it's a very big team, and the team, the project leader, Dr. Hegde, and the Shadi, and we have an advisor, Dr. Rawat, and these three people together with the team, they do. It's a zero surveillance project, mainly based on zero surveillance. So it's first the idea to handle and then move ahead with that. These are the number of extramural projects which India has. The number is shared, but of course at some point of time we had much more. So this is exactly what we have completed and what are ongoing. And this is a kind of the grants which we received through this EMR projects for NIAB. And these are the research publications and IP and the technology transfer. So if you just see to it, is a 3.04 average impact, which comes 3.60 for us. Some these are the PhD students or registering the Dumai or RCD or some other funds projects, and these are the only number of students with us currently at NIA. Some of the important government flagship projects also we have to handhold, and these are socio-economic upliftment activities for different farming communities, and there's a district at Yadgir which we have taken, and in fact we have developed a science museum wherein we have imparted quite a number of equipments from the institute. Idea is to create awareness and to handle the students. These are some of the activities wherein we were part of the Vigyan Sarvatra Pujate, Government of India program for 1 week long, which ended up having the National Science Day on 28th of February. And this is what Nia was participated with. The team really did very well. And these are some of the activities, as I said, that we are already with industry, either they are talking to them or we are already being translated for the incubators or maybe for the diagnostic. Some of the activities just which we did a day before to celebrate the International Women's Day out here at NIA. So this is what exactly, it was a science also awareness. We are not only creating awareness, we are very extending helping hand just to have the young ones in STEM. So this is all what the effort. We do invite them, encourage them, and we have a participatory approach of the scientists and the faculty in NIA. So scientific leadership, what NIA has showcased, especially in the COVID-related things, besides the pathogenomic, one has consortia, and all the the other strategies. To read all that and detail it out, but these are some of the scientific leadership we have just shown. And also, like we do have under the PM CARES fund, we do have a vaccine testing facility, which is very well placed, and the activities are going on in full swing at this particular VCS facility of NIA. With that, I thank everyone. The God is blessing me out not only during daytime, the evenings are equally present so that we venture into different areas where we are working. Thank you so much.
Thank you, Madam, for giving us a glimpse of the activities that we do at NIAB. And now we have, for the first time, we have respected Dr. Devanand sir, who is the director of IBRI. So we will be really willing to hear from him, and he is our guest of honor. So, Dr. Devanand sir. Okay. So, sir, over to you to get a quick remark on the program that we have, and please guide us that how should we plan and take it further. So, over to you, sir.
So, a very good morning to all of you. Dr. Vijay Barua sir, Honorable Chief Guest of the program. Dr. Subir S. Majumdar, former director NIAB. My colleague Dr. G. Taru Sharma, director NIAB. Dr. Dinakar Raj, my joint director colleague from IVRI, Dr. Sai Kumar. A vibrant team of the scientists from NIAB, dignitaries joined through this important video conferencing platform. Ladies and gentlemen, I am really grateful to my colleague Dr. G. Taru Sharma for providing this very important platform and sharing my views regarding a very important areas of research. So I will be just briefing you about just in brief, the status of the JEE in India and brief work done by the IVRI, and also the brief studies we have conducted in collaboration with the NIAB, and what would be the researchable issues way forward. Just in brief, I will be just touching. As we are aware that that total number of incidents on an average in India of JE is around 2,500 cases, and out of this, the 2,266 deaths due to JE virus we are getting annually, and that too, you see, the 80% of the total incidents are from Assam and UP. So this is the problems really hinting to all of us. And in brief, what the team of the scientists at IVRI, they are really working hard since long about the epidemiology studies of JE virus and developing the diagnostic tools for this very important disease. So our team, they were really did a good job, and they have come in a conclusion that more than 80% of the total JE burden in India is from UP and Assam. Till 2007, all the isolated JEV strain in India, they have also studied, they belong to genotype 3. But later on, outbreak of JE in Gorakhpur and UP and West Bengal, 2009 and 2010 respectively, revealed that the presence of the JE virus genotype 1. So these are the important lead which we had, and also our team, they have developed the recombinant protein based indirect IgG and IgM ELISA kits, which have been used extensively for screening of the swine's serum samples, which we are receiving from the different parts of the country. So this is that in brief our team did related to the epidemiology of JE virus and developing the diagnostic tools for this important JE virus. And some of the important study we have also conducted in collaboration with the INAAV. So the responsibility have been given to us for the genotyping studies, and what we have concluded that the genotype 3 of JEV is the dominating circulating genotype in the swine population in India. And second important, this lead, we have concluded that the high genetic similarity found at the nucleotide level between many of the JEV positive swine samples from our human isolates and our isolates from the positive isolates from the swine. It is clearly revealed that probable spillover of infections from swine to human. So these are the two important, and third important conclusion we have drawn, that there is a very close nucleotide identity between the JE positive swine sample and vaccine strain, which this is also indicating that the JE vaccination program in India must be continued with extensive coverage of vaccinations in disease-prone areas in order to control this important disease. And we have been given the responsibility of serosurveillance also. So this study revealed that we found that around our total samples studied, around 1,600 samples, we found that 22% of the seropositivity in this against the JE virus using this IgG and IgM diagnostic tools. We have also studied the risk analysis, risk factor analysis. This study revealed that this hygienic status of various pig farm are really involved in spreading of this disease, and also the waterlogging areas. Waterlogging areas is also a prominent factor for spreading this very important disease. In addition to this genotyping, serosurveillance, and risk analysis factor, we have also shared a couple of samples, positive and negative sample for with NIAV in order to study the RNA genotyping studies. So these are the, in brief, we have done at IVRI alone and in collaboration with NIAV. Really, based on the studies which we have conducted at IVRI and with the NIAV, the vaccines, development of vaccine is a key area. Although we have one vaccines in the name of Genovac, but it has been found that that this vaccines fail to offer the complete protection against JEV genotype 1 infection. Further studies are required to prove the efficacy of current vaccines against different genotype of JE virus. So this is the important issue to be required to be proved. And the other important issue that since long we were we were in the impression that the genotype 3 of JV is the only circulating virus in human cases. But this in the last decade, genotype 1 have also been reported in human cases. However, the literature also revealed that that genotype 1 from swine in India, but this is the key question need to be addressed, whether type 1 of JE is transmitted directly to human from birds, migratory birds, through mosquitoes, or through swine. Because we have received some of the incidents at Bangladesh, wherein we have a very less number of pig. So this is the important issue that the key questions need to be addressed. And although this we have studied in past also host pathogen interactions, but future investigations are also required to understand virus host interaction also. And the treatment is also a very we don't have any this treatment, although number of trials have been conducted in the past, but we did not develop any this therapy as far as this treatment of this very important disease. So I think there is a need to develop some treatment plan in order to reduce the burden of this very important JE, this virus. And these are the important researchable issues related to vaccine, epidemiology, pathogenesis, and treatment. And the responsibility given to IVRI, I think in future also we will be try to deliver appropriately, and we are really glad to be with Dr. G. Taru, who is leading this very important organizations, and we will be glad to collaborate in many other research activities also. Although we had our collaboration with One Health programs, and we need to develop some of the collaborations in other important areas. So I am really grateful to the team of NIAB and Director NIAB and other dignitaries for giving this opportunity to share my views among the audience. Thank you very much.
Thank you very much, Triveni sir, for highlighting the important leads drawn from the studies carried out at the IVRI. And we will take a note of the suggestions given by you, and we'll be looking forward to incorporate in the future work that we are planning to do in this program. So thank you very much, sir, for giving us deep insight about different kind of work being done there. So now, moving ahead, I would like to invite Dr. Syed Faisal, who is now the project principal investigator and scientist here at NIAB, and to discuss about the progress and outcomes of the flagship project so far.
Respected Bajirwala sir, respected Bharat sir, Director Madam Subir sir, Siraj sir, Manjula ma'am, respected sir, Sai Kumar sir, Chandra Raj sir, Dr. Vijay Tari sir, and all the distinguished scientists, eminent scientists, and speakers who are running online, my colleagues, students, very good morning. I would like to thank everyone for agreeing to be part of this important workshop. And today I'm going to present the progress that has been made in the flagship project of NIAB, genomic assisted pathobiology to identify the novel targets for diagnosis and drafting intervention of Japanese encephalitis and leptospirosis. So this project was initiated by Dr. Ravi Nandam in collaboration with IVRI. Dr. Mani Bhanse is there, and Dr. Ravi and Dr. Mani is working on JE component, and I am working on leptospira component. So I am presenting the leptospira part, and Dr. Ravi and Dr. Mani will present the JE part. So starting with the brief introduction, we all know we are expert in the field. So leptospira is a zoonotic disease which is caused by gram-negative bacteria of parasitic leptospira species. The most common parasitic species leptospira enterogens. There are more than 250 serover which are host specific, like hardware infects cattle and Canicola infects dog. The current diagnostics which are available are this is macroscopic egg rotation test, which is a gold standard, and rapid diagnostics have been developed based on ELISA, lateral flow, or PCR based diagnostics. However, this diagnosis has no limitation, like MAT is cumbersome and time consuming method of diagnostics, and there is no diagnosis available which can diagnose disease at a very early stage, and when the leptospira is in the blood, and there is no diagnostic which can identify the serover also. If you go to treatment, they have very good antibiotics which are available for treatment. These antibiotics are effective if diagnosed at a very early stage, the leptospira in the blood. But as soon as leptospira enters the organs and damages the organs, antibiotic also may not be very effective, or maybe host may already got a may die. So, but they also have certain limitations that we have seen recently the cases of antibiotic resistance of leptospira, and there is a severe reaction which is called Danish receiver reaction. When leptospira gets killed by antibiotic, it is LPS molecule which is causing this reaction. The current vaccine is still vaccine which is used mainly in animals. We don't have any human vaccine. The limitation of vaccine is that it provides short immunity, serospecific immunity, and there is no, neutralizing immunity that when we take the vaccine, the bacteria itself colonizes kidney and shed in the urine, where it becomes source infection for the other, other potential hosts. So, okay. So the current challenges are that due to the late diagnosis, antibiotics become ineffective when leptospira enters the vital bubble. So it can be only effective when you diagnose very early stage. So the aim is to identify characterize the pathogenic factors that are expressed in very early stage. If you identify those factors, then they could be targeted for developing diagnostics. So whatever diagnostic we are having is antibody based, when antibody already developed. So it's trying to switch from antibody to antigen-based diagnostic that is required in the field to identify very early stage. Second challenge is that the critical factors which are involved in from the host side for pathogenesis and the factors from the pathogen side and factors from the host side involved in defense has not been very well characterized or identified in leptospira. So now it is need to identify those critical factors and also the correlates of the immune protection. This will give a target for developing a new novel method like a diagnostic strategy and also for prophylactic or therapeutic. The current vaccines, which are limitations of short-term immunity and serospecific protection, that can also be overcome if we try to develop vaccines and targets which are identified through better understanding of how leptospira interact with the host. So it is very important to study leptospira host interaction. Several studies have recently shown that why it is very important. There's several interesting findings that come out. I just want to highlight a few findings where leptospira is known to colonize the kidney. But how is it very successfully established this renal chronicity? We know that kidney is a immunoprivileged site, so it might be a place that high leptospira. But how establishing that thing? A recent study has shown that a cytokine IL-10 is very important. So mice usually do not exhibit disease, but it could colonize in the kidney. So IL-10 knockout mice do not exhibit renal colonization. So one single cytokine, is not available. So leptospira could not colonize the kidney. And leptospira also has shown to upregulate the expression of IL-10. So this is a very interesting observation. So we can argue that based on this observation that upregulation of IL-10 would, suppress the interferon gamma expression, which would limit bacterial clearance and also favor renal colonization. So the antagonist, so this IL-10 is single cytokine has made an effect. So this is the identification, where this observation was made and this host factor IL-10 was found to be very important. Second observation is that leptospirosis prevalence is more in male than female, particularly the acute form of pulmonary hemorrhage. So this interesting finding was there in the study done in hamster model, where female hamsters were shown to upregulate the nitric oxide synthase gene. So the nitric oxide synthase gene was somehow associated with the inability of leptospira to survive in the host. So this could be this gene, this factor could be a beneficial factor protecting the female for pulmonary hemorrhage. So this is a very interesting finding just out of a single study. So it's time now to study this leptospira host infection more broader way, where several aspects of host and other can be covered in a single platform. And we can understand that there could be a possibility that there could be several factors from both pathogen and host side that could be involved in both pathogenesis and host defense. So omics is the one technology which has given this speed of how better we can understand how leptospira interact with the host. Omics, which covers the changes in the level of genes, the genome level when there is leptospira interacts or bacteria interacts with the host, and also at transcript level, at a whole transcriptome. At protein level, the changes of proteome and even the metabolites, when they change, when the pathogen interacts with the host. These changes in all different levels can be analyzed, and using various bioinformatic tools and softwares, we can analyze those changes and can narrow down to identify some critical factors or candidate biomarkers that can be exploited for developing new novel method of diagnostics or therapeutic intervention of vaccines. So this is the main aim of this project, where we are trying to understand how leptospira interacts with the host, exploiting this omics technology using genomics, transcriptomics, and proteomics, and then using various bioinformatic tools, we aim to identify those critical targets or for developing these diagnostics or vaccines. So this project has three objectives. Objective 1 deals with the diversity analysis of whole genome to understand the transmission events of disease, both leptospirosis and Japanese encephalitis, where we aim to sequence the genome of the bacteria in this platform and do diversity analysis. Second objective was to understand the genetic and molecular basis of disease pathogenesis to identify the critical factors of both host and pathogen leading to occurrence or non-occurrence of disease, both leptospirosis and Japanese encephalitis in various hosts. Here we aim to acquire this homocytology and try to identify what are the changes in both the protein and gene level in both host and pathogen. Third aim objective was to narrow down using the bioinformatics tools. Can we narrow down to whatever changes we observed in the whole genome or proteome or transcript or proteome level to narrow down to a few critical factors which can then be used to develop a novel diagnostic tools or a therapeutic or intervention. So in objective 1 part where we aim to do the whole genome sequencing of the leptospira isolates, coming from various sources like human or animals. This was the aim where we made several efforts to to collect samples and initiating new collaboration. So we got a few samples from Professor Nagmani from Gandhi Hospital. Actually, she could not come to this workshop with some commitment and last moment. She provided clinical samples where we tried to assess bacteria, but we could not assess bacteria because the patients have been given the antibiotics. So it's very important to see that we can get samples and before the antibiotic treatment. But however, we could get some samples and we could do some home study, which is in process of analysis. Dr. Balamurgan from Nevedi, he was part of this workshop, and he has agreed to provide samples from animal side. And Professor Burman from Assam has also agreed to provide some samples from animal to human, where this screening is going on. So once we get the samples, the aim is to do the diversity analysis to compare the zeros which are present in both the human and livestock, and what the difference in those genome and protein level, and then also to see that in this post adaptation whether the pathogen is losing any specific gene or there's an addition of any gene. In objective 2, we plan to study the post-pathogen interaction through omics. Where we initiated this study with a initial standardization techniques of omics of interaction of leptospira with human monocyte. We converted this monocyte cell line to the macrophages and then infected the leptospira and did the whole transcriptomics, proteomics, and then did the conjugate analysis of both the data to identify those critical factors using various software. I'm not going into that details what exactly we did, and this was done in collaboration with Dr. Sandeep Kurpara. I just want to give you just a few highlights of the data we got from this study from Pathogen side. But when we did the whole transcriptome or proteome analysis, we got several genes were differentially regulated, both upregulated and downregulated. Mainly the genes involved in the coding for surface proteins and cytoplasmic proteins. And then we did protein analysis, a lot of genes correlated to the transcriptome data, where we found upregulation, the green part, and also downregulation of several proteins, the mainly proteins of outer membrane and also cytoplasmic protein. When we did the conjugate analysis of both the data, we could narrow down to 16 genes and proteins which were come out and that were part of the relevant factors like outer membrane or the proteins involved attachment to the host cell. So these are novel proteins which we have found and we are in the process of validating it. From the host side also, we did a similar analysis where host side also we did the whole transcriptome and proteomics where we found several genes were upregulated and downregulated. In proteomics also, this lot of proteins were upregulated and downregulated. I will not go into the details of these proteins, but just want to highlight the key part where when we did the conjugate analysis, we could narrow down to 58 genes of protein from both sides. So 16 genes of protein from pathogen side and 58 genes from the host side, which was differentially regulated to be highly upregulated or highly downregulated during infection with the host. So the aim was this study was done on cell line just to establish technique or to the standardized technique. But major aim was to do the similar study in the clinical samples. So we wanted to do the clinical samples. When we got the clinical samples from the Professor Nagmani from Gandhi Hospital, we screened the samples and screening was actually done over there also, and we confirmed that out of 90 samples we received 25 were ELISA positive and 16 were both ELISA and RT-PCR positive. So those samples only which are both positive we further screened and we do not do analysis for all of them, considering the cost. But the best sample we have taken and we have done, we are doing transcriptomic analysis, and proteomic data is also being is process of being analyzed. So parallelly, we also started to bring interaction of leptospira with the animal host. We do the bovine model, where bovine macrophages get infected leptospira and did proteomic analysis, where we find at different time points differential protein expression, and in the 48 proteins that commonly express all time points. And when we analyze this protein, we found that a lot of these proteins were some of the known viral factors, some of the novel factors which are expressed various structures proteins which are involved in interaction with the host. So some further analysis going on, and we are trying to do transcriptomics, and that is also going on. In parallel, we also wanted to do this interaction study in the in vivo. So hamster is a very good model of leptospirosis. Then we infected hamster with leptospira, and then collected lung, liver tissue, liver, kidney at different time points. We did the analysis of lung only because we want to develop an acute model of leptospirosis, protein hemorrhage model, where we could see after infection there is severe protein hemorrhage in the animals, and there is significant body loss in body weight. We also analyzed the leptospira could detect in the tissue of lung, which you can see, liver RT with PCR from the lung tissue, to confirm that the lungs are heavily infected with leptospira. Then we did the proteomics analysis, where we could see in the lungs differential protein, of either side, up regulation and down regulation, where we are doing some pathway analysis, which proteins are interacting with different, host protein, and all this analysis is already going on, and this is the part of data. So objective 3 was the part where we want to validate. So we had a initial plan was that once we get a target antigen, so we will try to verify that target antigen. Based on that, most of the surface proteins are the viral factors. So if we can identify some new viral factors, we can first verify them. And further aim is to see that the antibody raised against this protein can neutralize the bacteria. We can also test in vivo, where immunization with this protein can induce protection against challenge, as vaccine candidate. Or also, if this protein is very, very important, then knocking out the gene can make the leptospira attenuated or lose ability to cause disease. So that also is the plan, where we identify some new factors, we'll try to make a mutant as a light into vaccine. Then our ultimate plan is to develop a rapid, early diagnostics. If we identify some proteins or genes which are expressed at a very early stage, then we are planning to develop a lateral flow diagnostic, rapid diagnostic in collaboration with Dr. Pankaj. So current progress in this is that the 16 genes we identified, we confirmed with RT-PCR, we found that these proteins were, some of them were highly upregulated and some downregulated, and we saw these proteins, we started cloning and testing and purifying. So that's all from my side, and I would like to thank all my lab members, especially the group who is working on this project, Vivek, Sridhar, Pallavi, and Vishal. Ajay and Pages are working on different aspects of Spara. Mohammed and Himali working on Activate. And director and IB, my scientist colleagues, staff are ready for all the support, funding from BBP. Thank you.
Ma'am, will we have some discussion right now or at the end?
Thank you very much.
Thank you, Dr. Faisal, for giving us detailed information about the progress of work towards leptospira. Now, Dr. Ravi sir is here, so I request Dr. Ravi sir for giving highlights of the work we've done for JEE. If Dr. Himani is available, is it possible, Dr. Himani, to make a presentation right now? He is not. Dr. Himani, can you hear us?
Yes, sir. But your voice is not clear, sir. It's not very audible.
Oh, great. So, good morning, Dr. Himani. There is some technical issue with the presentation from Dr. Ravi, sir. So, is it possible for you to make a presentation ahead of that?
Yes, sir. Yes, sir.
Okay, that's all.
I in my presentation, sir.
Okay, great. So, please, you can share your slides and then you will have your presentation now.
Okay, sir.
Okay, thank you very much.
Thank you, sir. Sir, my slides are visible?
Yes, Himani. Yes.
Okay, sir. Thank you, sir. Very warm good morning to all of you. Respected Director NIB, respected Bajar Barwa sir, respected Director IVRI and all the other dignitaries present there. A very warm good morning to all of you. I'll be presenting the work which is being done under the DBT sponsored project, that is genomics assisted pathobiology to identify novel targets for diagnosis and therapeutic interventions of Japanese encephalitis and leptospirosis in IVRI. Sir, the approved objective of the project for IVRI was to assess the circulating genotype of Japanese encephalitis virus in swine population of India. A bit of preamble. Why this objective was chosen? Because we know like Japanese encephalitis is a re-emerging viral zoonotic disease which is responsible for causing hundreds of children death every year in our country. And actual number of JE cases are much higher than being reported. Pig, being an amplifier host for Japanese encephalitis virus, play a very important role in its epidemiology. Besides being an amplifier host, pig is also the most practical sentinel animal to predict the human outbreaks of JAV, as it seroconverts 2 to 3 weeks before the infection occurs in humans. So if we can detect the JAV virus timely in pigs, we can prevent the outbreaks in humans, and it is a proven fact that surveillance studies in swine population aids in estimating the burden and forecasting the outbreaks of Japanese encephalitis virus in humans. As far as the JAV isolates from swine population of India is concerned, they are very limited. When we started this work in 2019, only 2 isolates of Japanese encephalitis virus were reported from India, the first one in 1986, and there was no other information available regarding that isolate, whether it is of genotype 1 or 3 or belong to any other genotype. Then recently from IVRI in 2016, from Dr. Sai Kumar's lab, they isolated Japanese encephalitis virus from stillborn piglet, and after genotyping study, they found that it belonged to the genotype 3. As we all know, like there are 5 genotypes of Japanese encephalitis virus, but in India, in humans, earlier till 2009, only 3 was there, which was reported from the human outbreaks. But after 2009, we started getting reports of 1 also. But in pigs, till 2019, only genotype 3 was reported.
So there were several researchable issues. One was that from pigs in India, only genotype 3 was there reported, but in humans we were getting both genotype 1 and 3. So the major key question was that when we started the, work under this project was that whether the genotype 1 of JEV is circulating in swine population or it is being directly transmitted to humans from ardid birds via mosquitoes, and pigs are not playing any role. So just to look at that, we thought of screening the samples from different parts of the country to identify the genotype which is circulating in swine population of India. Apart from that, the other issue was that recently in 2019, again a paper came and followed by several other research where they have proven like all the vaccines which are being used to immunize the humans across the world, they contain the genotype 3 strain. However, these recent studies, they have proven that these vaccines which are containing the genotype 3 strain, they are not providing the protective efficacy, 100% protective efficacy. In case of mice model, they have studied when they infected the mice with genotype 1. The protective efficacy was somewhere around 60% only. Thus, it became imperative to know the circulating genotype in endemic areas to understand the epidemiology of the disease and to design the future vaccination strategies for humans in our country also and in other endemic countries of the world also. So based on these researchable issues and key questions, we selected 4 different areas under this project to work on. First, we selected Assam. Why Assam? Because the maximum number of cases in last decade, they are being reported from Assam, followed by Uttar Pradesh. And here I have mentioned the cases which were reported in 2019. So in 2019 itself, Assam have reported 642 Japanese encephalitis cases, contributing to 25% of total share which were reported from India, followed by Uttar Pradesh, where 235 cases were reported, leading to 21 deaths of the children. So Assam from northeastern zone we have chosen, then Uttar Pradesh, because these two states report maximum number of JE cases. Apart from that, Assam has also maximum swine population in our country. It is around 2.1 million. And then we thought of choosing 2 other states from different zones. So from western zone, we chose Maharashtra because in 2019, 35 cases of JE were reported from Maharashtra, leading to 10 deaths, while from Gujarat and Goa, negligible cases were there. So from western zone, we chose Maharashtra. And since in 2019, the cases were coming in Haridwar and other parts of Uttarakhand of Japanese encephalitis. Earlier, the cases of JE were not reported from Uttarakhand or Himachal, but in 2019 they also started reporting cases. So from North Temperate Zone, we chose Uttarakhand. So we did sample collection from these different 4 states, and in total in last 3 years, we were able to collect 1,652 samples. And initially, sir, we collected both serum and blood samples, but after doing study, initially we found that even there was no difference when we were choosing either blood or serum in the detection sensitivity. Like matrix was not making much difference. Either we were using serum or blood, the diagnostic sensitivity was same only. And we published this paper. So after screening initial of 734 samples, later on we continued our work with serum sample only. Like for molecular analysis also and for serological work also, we selected serum sample only. And this is the flow chart explaining the processing of samples. Like we processed our samples for molecular study to know the genotype, and simultaneously we also did the serosurveillance using the IgM and IgG ELISAs which were developed by IVRI. And for genotypic study, we targeted envelope gene also, and for CPRM gene also. At the same time, we thought like better to quantify the viral copy number. So we did real-time RT-PCR also. Ultimately, for genotyping study, we did phylogenetic analysis based on envelope and CPRM gene of Japanese encephalitis virus. And using this phylogenetic tree, we assessed the circulating genotype in swine population of India. So in total, we screened 1,652 swine serum samples from 4 different states belonging to 4 different zones of India. and we found that the positivity using Tachman real-time RT-PCR was 35.97%, as analytical sensitivity of this Tachman real-time RT-PCR was up to 1 copy number, while using conventional RT-PCR, we were getting very less number, it was 3.27% because of its low analytical sensitivity. And at the same time, we did nested RT-PCR because we wanted to sequence our samples, because we can't go ahead with the we can't do sequencing with the real-time RT-PCR products. So we did nested RT-PCR for sequencing, and using nested RT-PCR, we got positivity of 7.69%. Overall, we'll recommend, like, if you are studying the molecular epidemiology of Japanese encephalitis virus, it's always good to use real-time RT-PCR so that the actual picture is being revealed because of its high sensitivity. And in our samples, we found that copy number was varying from 10 raised to the power 6 to 1 copy number per reaction, and maximum samples around 88 were having the less copy number, that is less than 1000 per reaction. That's why that might be one of the reason, like, the conventional RT-PCR and nested RT-PCR were not able to detect positivity in those samples. Overall, we were able to sequence 42 JEV positive samples. Maximum samples were from Assam, and we submitted the sequence with the GenBank and got the accession numbers. As I told before, like, we did phylogenetic analysis based on 2 genes. The first one was the envelope gene, and we got 2 sequences positive with this envelope gene. And the important thing which we found that our sequence, which we isolated from pig, it is AGE2, it was sharing the common clade with the human isolates, while the other sequence, again from Assam, it was sharing a common clade with the sequence which was earlier reported from IVRI. So this sequence revealed the spillover of infection from pigs to the humans or vice versa. And then other 40 sequences, we sequenced for CPRM gene of Japanese encephalitis virus, and we constructed the phylogenetic tree using a maximum likelihood method, and we got few important leads when we constructed this phylogenetic tree. The first was like, for those 2 sequences which we did for envelope gene, after phylogenetic tree was constructed, we found that those 2 isolates belong to genotype 3. Again, after constructing this phylogenetic tree for CPRM gene, for 40 sequences, we found all the 40 sequences, they belong to the genotype 3 of Japanese encephalitis virus. So we came to the conclusion, like, in India, genotype 3 is still dominant in the swine population of our country. And we got few of the important leads, like here, the sequences which are shown in blue color, they belong to our project. And we found that most of our sequences, they were sharing the common clade with the human isolates, revealing the spillover of infection. And a few of the sequences, they were also sharing the common clade with the neighboring countries like Nepal and China, revealing the spread of infection across the countries, maybe through the migratory birds. And we also found that there was high nucleotide identity with the sequence of our isolates and with that of the SA1414 vaccine, which is being used in our country for vaccination apart from Genvac, revealing that still we can continue with this vaccine in our country. The only thing which we need is the vaccination coverage should be more. So again, the inference drawn was the Japanese encephalitis genotype 3 was found still dominating in India, in contrast to the belief from other Asian countries where they are finding like G1 is dominating and it is replacing G3. But in our country, we observed still G3 is there. And this is ours was the first study where we revealed the JEV genotype in swine population from Maharashtra and from the north temperate region of Uttarakhand, which revealed circulation of genotype 3. Then spread of genotype 3 in swine population across the states of country, across the states of India. Like there was a high genetic similarity between the isolates which we were getting from Assam, Uttar Pradesh, and Uttarakhand. Then, since there was high genetic similarity between the positive samples of isolated from swine and humans and mosquitoes, so it revealed the spillover of infection of JEV from pigs to humans. Then dominance of G3 linked to the historical spread of JE outbreak from neighboring countries because we found high nucleotide similarity with the isolates from the neighboring countries, and the spread might be because of the migratory birds. Then ongoing JV vaccination in India, which is using the genotype 3 strain, may be continued with the extensive coverage. So the first part was it was the genotypic study, and the objective which was given to IVRI was to assess the circulating genotype. So we found that genotype 3, we sequenced 42 sequences, and we found genotype 3 still dominating in our country. Apart from this genotyping study, we also did serosurveillance on the samples which we collected. So we collected altogether 1,652 samples, and out of them 19% were positive for IgM, 14% were positive for IgG antibodies against Japanese encephalitis virus, and overall seropositivity of JE virus in swine population of India was 22%. Besides conducting this genotypic study and serosurveillance study, we also thought to conduct a small risk factor analysis study in Assam. And why we chose Assam, like as I told before, like maximum number of cases are being reported from Assam. So we thought of conducting this risk factor analysis study in Assam under this project. And in Assam, the population is mainly dependent on agriculture. Moreover, we have highest pig population in Assam. In 20th livestock census, it is reported to be around 2.10 million, and it is the only state where the population of pig has increased. If we compare with 90th livestock census, it has increased to over around 28%. So we conducted a risk factor study in Assam, taking into 2 districts of Assam, Barpeta and Sonitpur, from where the human cases were reported in the past. So in Barpeta and Sonitpur, we collected the swine blood samples from the owners and the information from the swine owners, and then we analyzed those 120 pig samples for the presence of JE virus and for the presence of antibodies against the JE virus, and the information was collected. Information related to risk factors were collected from these pig owners. So the questionnaire analysis, 16 explanatory variables which are related to the risk factor analysis were collected in the form of questionnaire. The outcome variable was JEV positive pig, and we were considering the pig positive either by serological or by the molecular test. So to assess the risk factors, we did descriptive analysis, we did multivariable analysis, and also the univariable analysis using the R statistical program. So coming to the results, the important findings which we found from our risk factor analysis was only 29% of pig owners were aware of JE infection in these 2 districts of Assam. And to our surprise, JE vaccination in children was not carried out in either district of the Assam, though both the districts have reported human cases of JE in the past. And we found that pig farms were present in the vicinity of 71.6% of households, revealing that pig farming is one of the main occupations in these targeted districts. Amongst the pig barns which we surveyed, we found that 78.3% were in poor hygienic condition, and waterlogging was also found around 160% of the farms. While doing univariable analysis, we found that 3 explanatory variables, including month, which is the monsoon season, and then the hygienic status of pig farm and water logging in the area, they were strongly associated with the JE positivity, and they were statistically significant also. Though other factors also, they were also associated with the JE positivity, like pigs staying in straw and thaw pig shelters, they were having larger odds of getting JE positive infection. And then distance of more than 15 meters between the pig shelter and owner's home has a protective effect against JE infection. But these 2 variables, they were not found statistically significant, though the other 3 variables like hygienic status of the farm, water lagging in the area, and month, they were found statistically significant and strongly associated with the JE positivity. Finally, on doing multivariable analysis, we found that monsoon season was associated with larger odds of pig getting infected with JE virus as compared to post-monsoon season, and water logging in the area near the pig shelter is also associated with the larger odds of pigs being infected with the JE virus infection as compared to the dry area. So these are few of the pictures which we clicked when we were doing the survey. Like in Assam, maximum people, they rear the pigs in the backyard. It is in very close proximity with the human dwellings. And they, in Assam, rice farming is also there. So it make a conducive condition for the JE virus to propagate. you can see the pigs are being raised in just behind the house, along with the other animals like poultry also. And waterlogging was seen in maximum houses because of the heavy monsoon conditions. So these all are the factors which make conducive condition for the mosquitoes to propagate, Culex mosquitoes to propagate, and ultimately leading to the JE cases. So inference of our risk factor study was that there is urgent need to conduct awareness program amongst pig owners along with the vaccination campaign for children against JE virus in the targeted district of Assam. Though vaccination is there in Assam, but when we took the feedback from the owners, they were telling their children are not vaccinated against JE. So maybe few of the districts are still missed. So there is urgent need. And if we see the case of Uttar Pradesh, recently earlier, the maximum cases were reported from Uttar Pradesh, followed by Assam. Now the case has reversed. Maximum cases are being reported from Assam, followed by Uttar Pradesh. So there was a report which reported that the cases of AES and JE in Uttar Pradesh during 2018 were drastically reduced due to massive intra-departmental preventive activities and awareness drives like Dastak, conducted by the state government. So such kind of awareness programs may be initiated in Assam also, so that we can save the lives of our children. Future perspective, we have conducted a small pilot study of risk factors in Assam, in 2 districts of Assam. Such kind of studies may be conducted in other endemic areas of India. There is scope to carry out further studies on molecular epidemiology of JEV in endemic states, focusing on circulating JEV genotypes. And there is need for a surveillance-based study to investigate the source of transmission of Japanese encephalitis virus genotype 1 in human JE cases. We need to know, we need to survey other states also to know whether genotype 1 is circulating in India or not, to design the future vaccination strategies for humans. Coming to the budget.
Dr. Himani, sorry to interrupt you. Now we are running short of time. So
Yes, sir, this is my last slide, sir. This is last slide.
Okay.
That is the budget allocated to IVRA. In last 3 years, we got 27 lakhs. And that's all, sir. Thank you.
Thank you very much.
Ravi sir, over to you.
Good morning, everyone. I'll be presenting the update on JE, what we have been doing, and this is not moving. Yeah. If you're talking about JE, already Himani has given an update of what JE is. So the major concern here in our study is to look at the amplifying host, the pig, and the dead end host, the humans, because it was really difficult for us to get the samples from horses. And this is the overlay of how the JE is distributed around the world. And we know that genotype 3 is predominant in the Indian continent, and genotype 1 is reported. And this mortality is around 25 to 30%. And there are permanent neuropsychiatric sequelae, which exist up to 50% in the adults. Now, the first thing that we wanted to do in this, as Sir has already briefed in his introduction, that we wanted to look at the global gene expression profile, transcriptome, and the proteome studies in JE. And under this, I was just looking at the literature. If you look at the literature, the entire literature covered mouse, humans, pigs, and all these were done as isolated studies. And there was no comparison. If you look at, no studies were conducted in the natural host, and there was no comparison across the hosts. So looking at this, we identified the gaps then, and no studies till date in the amplifier host, the pig, and the dead end host, the humans, and vis-a-vis the global gene expression profile and proteomics to explain differential pathobiology in the hosts. And also we wanted to look at the new concept of the role of epitranscriptome, the modifications in the RNA, vis-a-vis its influence on the pathobiology of the virus. So we undertook a study which is comprehensive, taking care of the transcriptome, the proteome, and the epitranscriptome to explain the differential pathobiology. And the major point here throughout the study, which we wanted to focus, is the pigs are amplified hosts and humans are dead-end hosts. And the objectives, as already projected by Faisal, this is the part, like the diversity analysis of the whole genome to understand the transmission events of JE, understanding the genetic and molecular basis of disease pathogenesis to identify critical factors that are mainly responsible for the differential pathogenesis in various hosts. So looking at the diversity analysis, we actually wanted to do this with the whole genome sequencing data, but we couldn't get the whole genome sequence data from the pigs because of the COVID. We couldn't get the samples, and the samples which went for sequencing did not give us good results. So immediately we had to shift for a different strategy where we looked at the genomic shift or the genotypic shift within the 1500 base pair envelope protein, where we found worldwide that there was a shift from genotype 3 to genotype 1. And the same trend was observed across all the countries across the world. And if you look at even in India, we have a shift here. Now slightly the genotype 1 is dominating. If you look at my introductory slide, we were pointing out at genotype 3, but here now slowly, as we are coming to 2020, like genotype 1 is dominating over genotype 3. We have done a network analysis to identify how the genotypes are varying across and how they are isolated across the subcontinents and different countries. And we have observed a distinct pattern just because of the geographical barriers. The mosquitoes couldn't travel probably, and the isolates are specific to India, and the isolates are specific to China, and likewise to other countries. And this second objective is to understand the genetic and molecular basis of disease pathogenesis. Under this, we wanted to have, again, the samples from live animals, and that was a hindrance mainly caused by COVID, and we had to shift to macrophage cell lines. And we concentrated on macrophage cell lines basically because the JEV hijacks the macrophage and then gets into the local lymph node and then into the spleen and then into the brain. So we thought we'll explore the transcriptome and also look at the epigenetic changes in THP1, which is a human cell line, and 3D431, which is a pig cell line. And THP1 is a monocyte cell line, which gets stimulated and gets converted into macrophage cell line, and 3D431 is a macrophage cell line. And in this, we subdivided the project into two different phases. One is the transcriptome in THP1. The other one is the epigenetics. Under this, we looked at the in vitro gene expression profiling of JEV, THP1, and 3D4 cell lines. And then we wanted to validate the determinants which we are going to identify. And the second one is the epigenetic changes in THP1 and 3D431. Under this, we wanted to identify the epigenetic modifications and the genomic regions associated with the epigenetic modifications and the role of these epigenetic modifications in JEV infection. Under the transcriptome in THP1 and 3D431, the work plan goes by to identify the gene expression profiling, further confirming the infection, and then sending the samples for transcriptome analysis. And the next objective is to validate these determinants which we are going to identify. So the preliminary study which we conducted, actually, even before getting into any detail, was to identify whether there is any dysregulation in the pattern of the proteins that are there when the samples are actually infected with JEV. Here, what we have identified is that THP1 infected at 24 hours and 48 hours. In both the cases, we observed that under gene ontology, the immunological processes were upregulated and immunological processes were highly enriched by the genes that are differentially expressed. This gave us a satisfaction that we are fine with whatever we are doing, and then we proceeded further, analyzing this data at 24 hours for the pig cell line as well. And the earlier one was for the human cell line, where we have observed again similar pattern, and the modulation of immune processes was also there under this study. And we went forward to compare the human cell line as well as the pig cell line here at 24 hours post-infection. And we have observed that the common genes that are involved, the DEPs that are involved, the common genes across both these species, there is a differential expression. If you look at the red one, it is a higher expression and the green one is a lower expression. And for the same genes in two different cell lines, the one is the pig cell line that is shown here, and the other one is the THP1 cell line that is shown here. There is a difference that is existent here as such in their expression. So again, we did a gene ontology for these common genes, and we identified that the immunological pathways are enriched. Then we did a pathway comparison, and we summarized saying that there was more negative regulation of viral processes in THP1 cell line, and the oxidative phosphorylation was enriched in the downregulated DEPs in 3D431 cell line. And then we compared the 24 hours and the 48 hours THP1, where we again concluded the involvement of apoptosis and involvement of autophagy. And then we looked at the viral processes, and we summarized that, okay, the cells that are infected with JE are now having certain variations that are because of JE. Having got these clues, when we presented the data in front of the committee, they said, like, what was the MOI that you infected with? And we infected with the one MOI there. And in fact, they said, like, we'll have to standardize the MOI. And then we standardized the MOI here. And at 24-hour DPI, HPI, like our infection, our post-infection, we found out that MOI 7 was considered for further study as far as the human cell line was concerned. And here, when we looked at the pig cell line, we considered MOI 1 as the final MOI for infection. Later on, they also asked us, like, we should also concentrate on the time point at which you need to collect the cells for transcriptome studies. And we looked at the expression of NS1 as well as NS3 gene expression. And we found out that 36 hours and 48 hours is the optimum time for the collection of samples post-infection. And this was further confirmed by the cytokine profiling in the THP1 cell line. And this further gave us a clue that 36 hours and 48 hours was the best time to collect the samples. Later on, we focused the same on the pig cell line, and we identified 48 hours and 36 hours again as the optimum time points to collect the cells for transcriptome studies. Having done all this, then we moved into a different, we covered this, and then we had something else in mind, and we thought epigenetic changes in the THP1 has to be focused on, and 3D431 cell line has to be considered for this. And then we moved into the epigenetic modifications, and we considered the major hypothesis behind this is the dimerization of the capsid protein with the histone and its impact on the numerous post-translational modifications that go by after the dimerization, right, would influence the viral replication and its life cycle. So under this, we had three objectives, wherein we looked at the identification of the epigenetic modifications we are looking at, and genomic regions associated with the epigenetic modifications and the epigenetic modifications in pathogenesis. We also looked at the histone modifications in 3D431 at 24 HPI. If you look at the quadrant 4 here, we have found that quite a number of cells are infected, and as well as they are having the expression or the modification for that particular. It is H3K4ME3, which is found in the quadrant 3 along with the NS1 expression. Coming to the work summary, this is what we are actually planning to do. And the major hindrance here is to get the transcriptome data that has gone by. For example, you can look at these samples which have been sent for transcriptome. They have been sent for transcriptome almost 8 months back, and we are still waiting for the data. And we have received only 18 samples, and we have to get another 6 samples. However, the samples are ready here. Totally 24 samples are ready for the proteomics data and epitranscriptome data to be analyzed, to be actually done. This protocol has to be standardized, and these samples have to be further run on the LCM MSMS to get the data. Thank you. Sir, actually what happens is when the samples go for sequencing, the RNA quality is checked, sir. And we again send the samples. That we cannot actually supratext.
Thank you, sir, for giving insight about the work being done by you on JEV and also by Dr. Himani. So now we have a fair idea what we have done so far and what, as per the objectives, how much progress we have made. Now we have our chief guest, Bejar Barua sir over here. So may I request sir to please give your insight about the work that we should take further, sir.
No slide. My slides will be here. Okay, little bit. Okay, very good morning. Almost we are in the afternoon after 10 minutes. Very distinguished director of NIAB, Dr. Mrs. M. Taru Sharma, the former director, a good friend of mine, Dr. Subir Majumdar, guest of honor, they are online, Dr. Triveni, the esteemed director of IVRI, my old friend and SAC member, Dinkaraj Ji, who is online. Here we have Director Meet, NRC Meet. We also welcome you. And then I understand we have with us also Manjula Sridharanji from University of Hyderabad. All distinguished scientists, students, and the participants who have joined online, ladies and gentlemen. Well, first of all, let me tell you, I am actually here to see Dr. Taru Sharma on the chair after Dr. Subir Majumdar. I have known Dr. Taru Sharma for about 20 years now. A person who has come up with her own caliber, a very good scientist. She is a fellow of many national academies, a lot many other things. We had the opportunity to interact with her, and over the years I have seen how she has grown. And here she is today. And Dr. Subir Majumdar, as you know, he has his own contribution in building up this institution and taking it to where it is today. We also have with us, of course, my neighbor. I almost forgot Dr. Khan from Librugar. I see him. So Subir Majumdar also has his own contribution. The most respected person across the country. Then when he was first director, then he had also his own contribution, particularly in selecting this particular or getting this particular land for this institute. So everybody, I am sure Dr. Taru Sharma will leave her own contribution after 5 years, because the kind of programs he has in mind, if when they will start going through, definitely, he will have another milestone achieved. I think the Dr. Subir Majumdar started with a genesis of this particular project. He said how it was rejected, how it was accepted later on. Let me tell you this rejection and acceptance of NIAB itself today. What had happened, incidentally, I was there when the NIAB EFC was presented to the then Secretary Expenditure Government of India in Delhi. That time I was working as Deputy DG, Animal Science ICAR. Prior to that, I had not known about this particular project. I had not taken interest also. One of my ADG colleagues, Dr. Rasul, he had taken a lot of interest. So I said in the first meeting, I said, since the meeting is there, I was to be away from Delhi. I said, Dr. Rasul, you go and attend the EFC meeting. But somehow that meeting got postponed. Second time when it was arranged, I was there, so I had gone there. Dr. Suraj Bhan, late Dr. Suraj Bhan, a great personality, I would say. He himself had come with his team to present the EFC. We had many discussions during the presentation. And since as I said, I did not take much interest earlier, that was the first day it was presented, and then a lot of interaction happened. And finally, it led to the rejection of the project in that particular format. Secretary Expenditure said, I'm sorry, Dr. Bhan, I think in the light of what has been discussed, this project needs a thorough review. All those kinds of things. The first project was, I still remember, it was around 540 crore project. Finally, it was modified this thing, and I think it got approved at around 200 plus crore rupees. So that is the rejection and acceptance, as you said in this project. I told you about the institute as a whole. Now the thing is, your program on genotic diseases, particularly the 5 diseases that have been mentioned, out of which you are discussing 2 diseases today. We have seen the kind of I think progress that have been made, both in Leptospira and JEV. I must compliment and congratulate the associated scientists who have so very nicely presented, the kind of overview Dr. Hegde gave first. It was so very good, followed by the presentation. Even Dr. Himani, I think, her presentation was good. Dr. Faisal, his presentation was, but all those presentations I have seen, the last one. Good progress thus far. How we go ahead, that's the kind of issue now. Gnosis, as you know, many things, but he has rightly said, Dr. Hegde, we all know today that around 60% of the known diseases, and more, alarmingly, 75% of the emerging diseases are of genotic nature. That's the thing. Emerging diseases that are coming, they are of genotic nature to the extent of 75%. EVAS had undertaken a study as to what is the economic loss due to genotic diseases in the world as a whole. And they came up with a figure that 100 billion plus US dollar is the loss due to the genotic diseases the world faces. And then they started studying as to what could be the reasons for the continued growth of the genotic diseases and almost unabated, migration of the diseases and all that. Then they could link it, the emergence of the diseases and all that, to the various human activities, which Dr. Hegde has also rightly said. The intensification of agriculture, for example, the infrastructure part, the extraction of oil natural resources, including the wild biodiversity that we have, all these have led. He also had mentioned about the travel these things, transportation and all that. Yes, but today there is no barrier. Earlier we had, today, one can move everywhere, anywhere, carrying the disease pathogens with them from one place to the other. All those things have happened, and considering this, WHO has constituted an expert committee. And that expert committee has to suggest the regions of emergence of the zoonotic diseases, all the possible causes, right from environment to the host to everything. They have to study expertly and give the suggestions to the world bodies like the FAO, the OIE, the WHO, even to the NHCP, the UNEP, United Nations Environment Program. All those, the expert committee is doing their job on this zoonotic diseases. Now, in the country, the Department of Animal Husbandry and Dairy, they have also launched this One Health program. One Health program they have launched, as you might be knowing, in association with the Bill and Melinda Gates Foundation and World Bank. So their target is to need all the stakeholders engaged in zoonotic disease research, and to find out an integrated solution to the zoonotic problem. So that's the kind of things. Now the thing is, at ground level, these things have happened. And these things are now to be supported, now to be backstopped by the scientists, not only in the form of developing diagnostic therapeutics and vaccines, but also in breaking the host pathogen linkage chain. That's the kind of expectation. Expectation is technology capsules from you, so that the other developments in addressing zoonotic diseases could be supported well. So that's how it's going. Now, out of the diseases, of course, the expert committee, they have been given the mandate basically for 5 diseases. The expert committee by WHO. In the 5 diseases, these are not covered as yet. Leptospira, JEB, and all that. 5 diseases are basically H5N1, the avian influenza, the Ebola, the Zika, and possibly the coronavirus. Now, as you know, corona is also now, it has been reported that it is also a disease of zoonotic nature. So when this can happen, obviously there is alarm. So that's how everybody is going or taking this particular aspect very, very seriously. Now on this thing, Leptospira and all that, we have been working, so you know better. Let me see my slides. Wow. You will not see, I'll see here. This is the old style of, the thing. We know in India, Leptospira is almost endemic in this part of the country, southern part of the country, the coastal region of the country. Basically, it is there, and the prevalence is much more. And we also have found, that has been rightly said, Leptospira antibodies, which is why antibody-based intervention we have been making so far. He has very rightly said, someone, I think last presenter, that antigenic-based approach we have to make now. We have been having so far this thing. So in this all geographic region, as you know, various Leptospira, serovars are prevalent, and they are associated with one or more maintenance hosts that we have already known that serve as reservoirs of the infection. Now the thing is, I'm very happy that antigenically related serovars, which are classified into, or they are basically classified into or more different species, and the serogroup is often found. It is often found in several species that you know definitely. The serover identification of isolate is essential. How much you are doing, I do not know. Serover identification, as we have known, identification of isolate is very essential so that we can understand the epidemiology of the disease. But because we have very few labs in the country to perform in a cross-agglutination absorption test, we are not in a position to go into that serover level. So I think one or two, attempt if you can make, that will be fine. We also know very well that leptospire, how it enters the host. Basically through skin and the mucous membrane. We all know. And then it gets circulated into the bloodstream, and then it dissipates to the different target organs, the liver, the lungs, and particularly the kidneys. In kidney, they are present for several months. Now the researchable issue here is what sort of managemental kind of technology we develop to see that this entry level through the skin, through the other things are to some extent controlled. How best we can do that? That is a managemental approach of trying to control and contain this particular thing. Then your scientific ways. See, this is there in the kidney, and they get said, of course, the pathogens and all that. When they are in the kidney, what sort of things we can, what kind of advanced tools and techniques we can utilize to see that we keep them fixed at the kidney level itself, so that it does not get set. What sort of things we can do, if we can do, that sort of a thing. Now, in this, I do not know whether you are using biofilm. These days, I am told biofilm is emerging as a very promising field in leptospira research. I did not see it whether you have used that, but as has been said, that it is a very promising field because it could give rise to the identification of several novel proteins. You have identified the novel proteins. I am so very happy that you have brought down the number of proteins, novel proteins, to 16. So this itself is a success. You can now work on 16 from the host, 58 from the, I think, 16 from the pathogen, 58, I think, from the host. You have brought it down to that level so that you can work in a very concerted manner. Now, so therefore, I think one of the things could be that if you, together with that, use also the biofilm technology, because through this you can also develop diagnostic tools. Many people are talking about diagnostic tools, therapeutics, and all that. What about developing some screening tools? So we hit on diagnostic tools. This is screening, like, we have a screening thing for, say, for example, which is tuberculosis, the kind of tuberculin test we call it. So whether we can, develop that kind of screening so that every 3 months or so, we screen the animal and develop the screening kit, give it to the farmers, the owners of the livestock, so that they themselves can screen it. In COVID time, we have seen, there were kits available. I could see myself whether I am positive or negative for this thing. So that kind of, screening kit, if you can develop, that will be very, very, encouraging a thing. And that kit, as I said, must go to each person, so that you cannot go there to each livestock, but the owners must know how to use them, utilize them, and report. Fortunately, in animal husbandry program, they have also taken one program on identification of animal. That identification thing they have, and it's sort of a kind of what is called Aadhaar card for animal. and the animal health cards sort of thing. So that we can, connect with each animal, and they get the data and analyze them in a common server, that kind of a thing, So therefore, try to see whether some screening, Bruxelles screening kit could be used, which should be very handy. Now there is yet another development. I think I have seen that a novel elastin binding protein of pathogenic leptospira, which may be promoting the attachment of leptospira to the host tissues. That has also been reported. So elastin binding, sort of this protein. And again, OMP-L37, you must have heard. Its use is also very, very good. It has been said that you can do that particularly to mediate host attachment, and this will also have potential role in clinical application. So that sort of a thing. Now, some of the things, whether you have done or not, I don't know. How to sort of how to study the sort of stage of leptospira binding with the extracellular matrix. How we study the different kind of stages, and what is the adhesion pattern of that. So that kind of a thing. And we have also to develop, I think, complete understanding of leptospira binding with proteoglycans. How is the binding like? How we can, this thing. So then cell interaction and adhesion of leptospira, you must have done that. And then interaction of leptospira with host fibrinolytic system. What sort of interaction they normally will have, how we can benefit from those kinds of interactions and all that. So, and then you are also, I think, using the tools, gene knockdown, maybe gene silencing and all that, where to do this, how to do this, all that kind of, all this kind of advancement that you have yourself achieved. And sort of recombinant protein, etc., that you have generated. I think they are now to be combined with the numerous data that already you have from different sources that you should combine. And one of the options rather should be to merge these adhesions as chimeric construct for development of a more viable, more efficient vaccine. Now, in leptospira, ICR Nivedi has done a lot of good work already since last so many years. They have been on this particular leptospira, which has been recognized also nationally and to some extent internationally. What is your tie-up with that institution? Where they are, where you are, how you can complement and supplement your effort, in achieving the objectives that you have laid for or set for yourselves. I think this type of an approach will definitely be very good, because they have also come out with diagnostic kits, Nivedi, to the extent I know. They have come out with diagnostic kits which have been accepted. So similarly, I think your institute has also developed that chip, which is for which you are well recognized. So, similarly, some other institutes are recognized for something else. So how we collaborate, go for it, and finally give benefit to the end users. That should be the attempt. On JEV, many things have already been said, and your progress so far is very good. Some JEV incidence is more, obviously, because the big population in northeastern region is highest. Northeastern region as a whole, it is highest. In some places, pigs are with the family. In some tribal houses, if you go, it is sort of, raised houses where the ground portion will be pigs and upper portion the human being will live. So they are in constant touch with pigs and all that. So here this thing is, say your vector is mosquito, your reservoir is the, I think, the kind of birds that you have, water birds particularly, and finally amplifier is pig. Now the thing is, this mosquito, birds, pig. Now you are focusing on pig, that is good because it has also the similarity with the human being. Now, if we can do this, I think your approach is right that you have taken pig as the model now that I have seen from your presentation. I think something Leptospira similarly we need, but I am worried. One person said, Dr. Triveni, director IVR, I said, there is a report of genotype 1 from Gorakhpur and West Bengal in 2009 and 10. But in the last presentation, I heard that genotype 1 has overtaken genotype 3. That is a matter of concern. Now, to what extent that is, how much that is true, has also to be seen. Has it really overtaken, or genotype 1 is restricted to some pockets only, like the UP or Assam? In JEV, the easier thing is the states are limited. 85% of the JEV infections are located in 5 states. And out of 5 states, 81%, as Dr. Triveni has said, out of 85%, 81% is in Assam and Uttar Pradesh. So therefore, focus should be there. Those are the sort of endemic areas. So you can get a lot many samples, all those kinds of things. Sorry, am I boring you? 15 minutes more. Yeah. I think the kind of work we are doing is fine. I think your project will come to an end after 6 months or so. I think you will need for some extension. I think going by the importance of this project. Otherwise, JEV, if you see, particularly JEV should not be a matter of much concern, because if you see a typical show, some 2,545 cases, if I remember well, 2,545 cases. That is, it is not an alarm, actually. There are 266 deaths. On footpath itself, so many people dies. But here the concern is the death of particularly the children. The concern is, that is why it has this thing. But you have every possibility or probability to contain and control JEV much more easily than your leptospira, because of the 5 state location against around 23 states where leptospira is prevalent. So that's why, I'm sure the kind of work we are doing are definitely fine. I compliment and congratulate Dr. Subir that time and all the team for finally bagging this project. And the country is looking forward to your results. It could be your, as I said, a screening kit, could be a diagnostic kit, could be a vaccine. They are all looking forward to it, and I hope you will fulfill the expectation of the people and backstop the effort of the WHO as well as Government of India in containing and managing the zoonotic diseases, particularly in India. We have to talk for the globe as a whole, but at least in India, if we can do that, that will be fine. Thank you very much. I must thank Dr. Mrs. Taru Sharma for giving me this opportunity to be with you. And this institute, as I said, I was associated right from its birth. You have said discussion said like this. Then I knew a little bit about your programs when I was there in what is called RAC. And I'm sure with the kind of background each scientist has, the future of science in this institute under the leadership of Dr. Tarun Sharma is going to be very, very bright. Thank you.
Thank you very much, sir, for your insightful talk, and you have given really a good suggestions. Having tremendous experience in the field, the kind of suggestions you have given, we have never thought of, and we have been very positively taken your suggestion. And sir, we want to assure you that we will work in the way that you have suggested. Something we are already doing, like I want to highlight that you mentioned about the proteins which bind to extracellular matrix, the ECM component, adenosines. So that is our focus, sir, and your suggestion for collaborating with the Nivedi. So the Nivedi, ICR Nivedi and IVRI has done a tremendous job in field of leptospirosis. They are the pioneers. So I was fortunate enough to collaborate with Dr. Bala Murugan, who is also in the the panel. So with his, I think collaboration, I think I will be able to get some success. And I think we as a youngsters have to learn a lot from tremendous experience you are having. And we need expert advice for all the people who are over here. So we will follow your direction that we want to assure you. So moving forward, I think there was a tea break. Tea break is there, but I just want to, because we are running late, so people can grab tea and can come back. We can start the session, next session. Before starting the session, I just want to formally welcome Dr. Vijay Achari, sir, who is Director of RMRC Portware. Sir, you are online?
Yes, I'm online, please.
Yeah, sir. So very, very welcome, sir. Just thank you so much for agreeing to be part of this workshop. I just want to express that Vijay Achari, sir, has been very helpful in helping me establish leptospira research in NIAB, and I have visited him, Port Blair physically 2 times. Visit that place, and sir, you also need your support in future also. So we will looking forward to your talk, sir.
Thank you very much for invitation. I'm grateful to you and your director, all other colleagues or experts gathered there. My sincere thanks to them.
Thank you, sir. So I just moving forward. So actually, Manjula ma'am has some, for some personal reason, she actually, I'm very thankful to her. She has came over here and agreed to come physically and to deliver a talk. So ma'am, because Dinkar sir is there for giving a talk. Yeah, you can. Excuse me, Dinkar sir. Actually,
if you permit, madam's husband is unwell. So can she talk before you, if you are okay?
He can't. Hello, Dinkar sir.
Yeah, okay, sir. Okay, fine.
Yeah, thank you, sir.
Yeah, I'll wait.
With this, I request my Manjula ma'am, who is professor at University of Hyderabad, to please give her expert opinion. Thank you.
So while he's fixing the slides, I would like to thank the director, Dr. Tarun Sharma, then the former director, Dr. Subir Majumdar, our chief guest, the former vice chancellor of AAU, Dr. M. Bujja Barua, and other dignitaries. And of course, all my friends at NIAB, and especially Faisal, who has been after me to give this talk. I have chosen diagnosis. Yeah, this one, no? Yeah. I've been interested in the diagnosis component because, as we all know, I mean, the previous speakers have said quite a bit about the disease, so I probably would skip a few things, but mention some of the salient things. Like, we all know that it's a neglected disease. There has been less funding for this, and this has been deliberated also in the International Congress that was held at Andamans, coordinated by Dr. Vijayachari. And diagnosis is one of the challenges because of the lack of proper diagnostic tests, and it is often mistaken for some other infection, and therefore it sort of contributes to it being a neglected disease, and much needs to be done. And we all know the kind of reports that we have in India, especially in the coastal districts, which has been mentioned. So we know the number of deaths that was there in 2005. In Kerala, we have recently been hearing other coastal areas, Chennai, Gujarat, etc. We have this. And I'm not going into the points here because it has been dealt with. And essentially, this is to show that the humans are accidental hosts, and basically our human practices is going to contribute. So preventive measures also is going to be helpful in controlling this disease. And this is just to show you the various conditions, which shows that this is more of an occupational disease and seen in different tropical countries where it is referred to by various names. So we won't go too much into the causative organism. This is a SEM which was done by my student recently. And we all know the kind of the number of serovars, which has been traditionally done by serotyping. And we have also molecular methods, but and these are, I'm just highlighting that you cannot just detect them by using conventional staining techniques because of the fact that they are very, very thin, and conventional staining techniques will not help to identify. And what is done is dark field microscopy is used to detect the corkscrew movement, which is inherent of this spirochete by virtue of two internal flagella, which runs from the ends to the inside, giving it the characteristic movement. But again, we cannot use it for clinical material because of artifacts like fibril threads, etc. So even though some labs do say that they use dark field microscopy, it cannot be. It is not going to. It's going to give you false positives. So I'm not. This is some of the things which we have. Actually, it's a video. I don't think I'm going to tell it to this audience. Is very familiar with that. But basically, I just want to show that here you have the disease often is self-limiting in the sense that most of the patients are going to recover because our immune system is going to take care of that. But it is going to be causing a high level of morbidity and therefore a lot of economic implications for the working class. So therefore, it's important that we sort of have some diagnostic tests for this disease. And you have the risk of some of the, a small population or a group or 10% of the patients who can develop the severe form called as the Whale syndrome, where the hemorrhagic manifestations are going to be a cause of great concern. And this can often be fatal. And we can have the situations where the hemorrhage can be in the lungs. And this has been seen in a lot of cases in the Andamans. And we have the liver getting affected with jaundice, again, mistaken for, non-leptospira cause for jaundice. And then renal failure is something which is, again, known, but again, it is not diagnosed as due to leptospirosis. So even in the animals, so this is as far as the humans are concerned, but in the animals also there is a huge amount of economic implications. So here, diagnosis, control measures will include diagnosis, treatment, and the prevention. I'm not going into the details about the treatment and the prevention, but I'm going to focus quite a bit on timely and specific diagnosis. And it is a well-known fact that there is a gross underreporting of cases. And treatment, of course, is very effective using penicillin and doxycycline. Prevention, as I mentioned, prevention using protective clothing, etc. We are still away from getting a proper vaccine, though some countries, they say that they do use some animal vaccines. So this one is just to, this has already been mentioned that the pathogen enters through the skin. It's going to remain in the circulation. I just want to mention this so that it will decide the kind of the diagnostic test that we would use. So it remains in the circulation. And the moment the antibodies come, the LPS is going to be highly antigenic. Therefore, the first antibodies will be against the highly immunogenic LPS. So once antibodies come, the organisms will disappear from the circulation into the tissues. And as was already been mentioned, so they prefer to go to the kidneys and they're going to remain there. They can multiply there. And the point to note is that they are going to be continuously, they're going to be shed. I won't say continuously, they are intermittent shedders. There are periods when they'll be shed, and then there can be times when there is no shedding at all. And this is very common in the case of the animals which serve as carriers. So this is going to get reflected in the leptospiremic phase, where if you're going to look for the organism, you will have to look at it only in the first week or so. Following which, we'll have to look for the antibodies. The humoral immunity is very strong. Therefore, screening for antibodies against some of the left cell proteins is going to be the one that I'm going to focus on. So therefore, if you look at what are the kinds of tests that we can do, so the first thing is, clinical presentations is not going to help us because it does not provide a differential diagnosis, because the symptoms that is seen is also seen in a number of other infections, which can be viral as indicated here, or it can be malaria. All these are prevalent in tropical countries. And also the scrub typhus is another bacterial infection. So therefore, laboratory diagnosis is important. And timely diagnosis is what I'm going to emphasize. Timely diagnosis is important so that you prevent the progression of the disease to the severe form. That is number one. And also during monsoons, we hear so many deaths being reported. So if we have effective screening methods, that would help during the monitoring of the outbreaks. And of course, if epidemiological surveys and surveillance studies are being done, we need a proper diagnostic thing. So what are the tests that can be done? As I mentioned, like leptospiremic phase, the logical thing would be, can we do culture, like as is done in either bacterial infection? But no, these do not grow very easily, obviously, because, possibly the patient is taking some kind of, other antibiotics, etc. But even otherwise, it's a little bit fastidious. So culture is not conventionally done. PCR, definitely yes. Now it is being done. I think Dr. Vidyachari will be talking about that as well, because they do quite a bit in their thing. But what I'm going to sort of focus is the immunological, the immune response, where the MAT is the gold standard. And we also have commercial kits. The example is PanBio ELISA. Now I'm just mentioning this to highlight that, okay, the MAT test is basically, I'm not going to go through the test, but what I want to emphasize is that a patient's serum is going to be serially diluted, but we are going to need live serovars for this, and agglutination is going to be detected like this. So there is going to be, and this is seen only by under the dark field microscopy. So this is going to require the culturing and the maintenance of serovars, and since it is going to be detecting the anti-LPS antibodies, we need a panel of serovars. So conventionally, the ideal way would be to have as many as 30 serovars, even though many labs would do something like 15 serovars, but it's very expensive to maintain this. So maintenance of the live serovars, technical expertise does not allow it to be used in any routine laboratory. So therefore, we need a economical and easy to perform diagnostic test. So as I already mentioned, the demerits of the MAT, even though it is highly specific, It is a gold standard. It still suffers from some of this. And the commercial tests are all imported, so economical reason. So therefore, we need a simple indigenously developed ELISA-based system. And if we want to do that, as has already been mentioned, we have, you can have the basal levels of antibodies are also going to be high. And also the non-pathogenic ones, the Leptospira biflexa, is there in water everywhere. So you can have cross-reacting antigens. So you need to have a specific antigen that we need to detect. And this is where we have come up from our lab with two antigens, that is the human binding protein and sphingomyelinase as diagnostic markers, where we made a transition from basic research. Essentially, my lab has been focusing on understanding iron acquisition, as well as looking at the virulence factors, sphingomyelinases, which are responsible for the hemorrhagic manifestation that is seen in this disease. So we've been using these as diagnostic antigens. And the reason why we have chosen is that they are pathogen specific, they are expressed in vivo, we have high levels of antibodies in patients with leptospirosis, and the respective antigens were effectively used in an ELISA format. And it seemed shown to be even superior than the MAT when we compared both the human samples as well as the bovine samples. So I think since I don't have a lot of time, I just will sort of mention that the human binding protein is basically an outer membrane protein that plays a role in uptake of iron from human. And this is going to be a typical the membrane structure of the gram negative, which is very similar to what is seen even in the Leptospira. And we are the first people to have identified this HBPA and characterized this as a human binding protein. And what I want to emphasize in this gathering is the clinical significance of it. So this is just to show you that it is expressed by the low iron organisms when iron is limiting. It is not seen in the high iron. This is the control. This is confocal microscopy. But this is what I want to emphasize, that it is a diagnostic marker, and we have used it in several studies in leptospiral uveitis, which we did a collaborative study with Arvindai Hospital from Madurai. And we also used, for screening bovine samples, we compared it with two well-known antigens, LIPL32 and LIPL41. And then we have used it in a patient in Hyderabad by a clinician who used this assay. And also we have shown the urinary excretion, which I will talk about. So the urinary excretion, both of antibodies as well as the antigen. So this is the recombinant protein that we have made and used it in leptospiral uveitis. This is, again, as I mentioned, is a study with Arvindai Hospital. And you can see that we have chosen these group of patients with cataract controls and endemic normal serving as our controls. And we have the uveitis cases, and we have the systemic as also controls. And you can see the antibodies. So you definitely need to have a baseline control. It is not going to be zero. There has to be a baseline. And then you can see that these are going to be below that. And you can see that how the HBPA ELISA has been useful. And this was compared with clinical presentations as well as with the commercial PanBio. And it definitely has been useful in in screening of leptospiral uveitis. And there are and also we saw in our own this thing that it is better than the MAT, as also seen with LIPL41 and LIPL32 ELISA. This we have done with bovine sample. I'm not showing that over here. So therefore, we feel that this can be developed into a proper ELISA format. And also we have tested it on a lateral flow system for our HBPA. And you can see the kind of nice development with a positive sample. This is a negative sample with the control showing up the reading. So that could be used as a bedside test as well. And this is the sphingomyelinases. Once again, for paucity of time, I'm not going into the details about it, but I want to tell that we have several sphingomyelinases. But there is a common domain, which is the enzymatic domain over here. And we've used that domain, we've cloned that, and we've expressed it and used that for all our studies. And we've done a considerable amount of work on the structural determination and the biological activity of that. And you can see that different leptospira, the pathogen, this is expressed only by the pathogens, different leptospira, pathogenic enterovirus, they have different numbers of these expressed. And again, it is not known why they should express more than one and why 5 here, 3 here, and 7 here. This is again known. Is it the niche where they're going to be multiplying? Is that going to be influencing it? It is a possibility, but that further work remains to be done in that area. So here we want, and also we know that these toxins or the virulence factors are made as a pro molecule and they will be processed. They will undergo post-translational cleavage to give you a functional active molecule. And that is something which we were interested in looking at and which we identified and recently showed that we actually see a 42 kDa sphingomyelinase C that is expressed when we infect virus cells with the serovar Pomona. And we've done quite a bit of work on this. And this is just to show you the viability is, there is detachment of the cells. The viability is lost by flow cytometry. And this is a cell thing where you can actually see the holes or the perforations that are made by this when we use the live organisms. And if we use the culture filtrate and add it to pure cells, you can see the kind of the damage that is seen. And this is again to show the binding of the specific antibodies by confocal microscopy. We have shown that. And this is just to show you that there is hemolytic activity and of this. This is the hemolytic activity. And this is the 42 kDa band. This is the internal control, LPL32, which is used to show that we've loaded equal amount. So this is the cell, this is the virus cell derived, and this is the in vitro grown cultures. So you see that the 42 kDa is expressed only upon infection with virus cells. So, and this is just to show the biological activity. So we have to show the biological activity both by hemolytic activity as well as with the enzymatic activity. So this is more from the characterization of the molecule. But what is important for this point of thing is that we have shown that if we screen for the urinary antigens, both HBPA and sphingomyelinase, in addition, we have also used the LPL32 because that is the one which we know is a predominant antigen. And we were able to identify or differentiate leptospirosis patients from dengue patients. And this is a study which we did with a clinical group from Kerala. So we've got samples from them, and we have used the 3 groups of patients. This group is totally dengue, and we have done the screening by all our experiments. We have used MAT as a gold standard, irrespective of whether we were doing ELISA or not. So this is all MAT negative, and these patients who are leptopositive, they are MAT positive. And here we had 6 cases of pyrexia of unknown origin. And out of these 6, we will show you that 2 of them, we showed them or we established as leptopositive by looking at, first of all, we did antibodies in the serum we screened. And you can see that, and here, we use the non-pathogen Andamana, also whole cell sonicate of the pathogens like Pomona hydra bovis, as well as Andamana. And you can see that we have shown the antibodies in the serum as well as in the urine with our Andamana non-pathogens showing very low reactivity over here. And if you see the urinary antigens, it is so high over here. And we have looked at LPL32, the FLA-A, the LPL41, and our human binding protein, that is HBPA. And you can see that the controls are very well below the line. And these are the positive samples, and the arrows indicate 2 samples of the PUO, which is actually showing higher values, which we were able to show later on that in fact the urinary sphingomyelinase was also seen in these cases. And we also did a Western with the urine samples, and you can see the LPL32 that has come up in some of these samples that we've seen. And you can see the 42 kDa band. Additionally, we are also seeing in 2 cases 58 kDa. So we assume that, or we presume that there is possibly another sphingomyelinase that is being expressed, which is post-translational translationally, clean to give this, but we need to do further characterization to understand the nature of this, whether this is from a different sphingomyelins. As I mentioned, there are 5 sphingomyelins produced by the seroviral. But then our differential diagnosis was very clear that the parameter, you can see the serum antibodies in leptospirosis cases, dengue, and the urinary antigens, you were able to see this, they were absent in this, and therefore we were able to say with surety that these are the 2 PUO cases were definitely lepto and not dengue cases. So this is just to summarize the slide, that we can be using this as diagnostic antigens. Antigen detection is going to be even better, and this is non-invasive, and then it is going to reflect active infection as well. And also further study on these sphingomyelins will help us to understand better the host pathogen interactions and also identify these virulence factors, which are very, very important to understand the hemorrhage that is associated with leptospirosis. So of course, nothing goes without acknowledging the funding. So I had funding from DBT when I just started the work, quite a bit of funding from the defense, ICMR. And of course, it goes without saying that Dr. Vijayachari has been more of a friend and a colleague who has helped with establishment of my cultures, etc. And collaborators with Dr. Ratnam from Arvindai Hospital for this work, as well as Dr. Epan and Dr. Tresiyama from the hospital in Kerala. And if this is not possible without my workforce of PhD students, they have all completed, they are establishing on their own. She's still continuing with her work on leptospira. So they have done immense work, and I would like to thank them personally on this. Thank you.
With the permission of the chair, can I talk to the Manjula madam, please?
Yes.
Thank you very much, madam. You are elaboratively explained on all things. Your work is well known.
Yes.
So it is an opportunity for me to come Hyderabad and meet you, but unfortunately due to COVID, I could not come there. So okay. I would like to express my sincere thanks to you.
I was looking forward to meeting you.
As you know, madam, that COVID is only one lab. You have to take care about all the whole island. So the moment some people are restricting that all the things are there, that's why I could not come there.
Thank you.
So thank you. Next time I will see you. Certainly.
Thank you, sir. Thank you.
Thank you, ma'am. So we are running short of time. So I quickly invite Dr. Dinkar Raj sir to give his expert opinion. Dinkar sir.
Yeah. Yeah, salutation and greetings to this august audience, Dr. Bhujan Barwa sir, Dr. Subir and Dr. Taru Sharma, all the guest speakers and all the other scientists from NIAB. Thanks for the opportunity. So Dr. Manjula Sridharan has made my job that much easier and probably quicker. So I will probably finish it in lesser time than that was allotted. So I'll be discussing something on our experience with leptospirosis, including diagnosis and the challenges that we have faced over the years. So just to highlight one thing, the leptospira gives you a broad spectrum of clinical presentation. And why diagnosis becomes very important is because it drives the line of treatment. Many people with fever are given antibiotics without identifying the cause. And if the fever doesn't subside after 3-4 days, then only they start suspecting leptospirosis, and that is when the blood samples also are sent to our lab, by which time we could see antibodies, and very difficult to see the antigen or the leptospira in the blood sample. And that is, as you said, MAT is the gold standard, but it's quite cumbersome and
Our lab has been doing MAT for a very, very long time, maybe 30 years. One of the pioneering labs that started MAT in our college in the Department of Preventive Medicine. And now there is a separate lab called the Leptospira Research Lab, which is functioning at Madhavaram and which does diagnosis of human sera samples with MAT. And lots of kits, as Dr. Manjula said, are available, but let us discuss about the performance of those kits also. I'll skip this slide, but only to highlight that all the animals, humans, soil, and water are also involved in the pathogenesis of the disease. And again, about the organism, we'll skip. So in our Leptospira lab, we are maintaining about 12 serovars. Sero groups which we use in the MAT, the sero groups are identified here. And just the samples that we have analyzed during the last year, the year before 2019-20. If you see, these are the various tests we have done, DFM, dark field microscopy, MAT, ELISA, PCR, as well as culture with both human samples that are referred to us, as well as animal sample that are referred from the veterinary college hospital and also from across the state, as well as our own epidemiological studies. So you see the number of positivity is very, very low. Out of 1,478 samples tested during 2019-20,
Hello, sir.
positivity.
Hello, sir. Your slides are not visible. Please end the screen, end the slideshow. And then in the PowerPoint presentation, start slideshow in the PowerPoint presentation now. After starting presentation in the slideshow mode, and again start screen sharing, sir.
Screen sharing.
After starting slideshow mode, start screen sharing. While starting screen sharing, that two slides, second screen will be there, slideshow screen. That screen share, sir.
Can you see now, sir?
Yes, sir. Yes, sir. It is visible now.
Sorry. So, didn't you see from the beginning? Do I have to repeat?
Okay, sir. It is okay, sir.
Yeah, okay. Yeah, I was just telling these are the 12 serovars that we maintain in our Leptospira diagnostic lab. And we perform MAT on clinical samples, both human as well as animal samples. And during the year 2019-20, we find that the positivities are very, very low. So a human sample positive, we do different tests also, dark field, MAT, PCR, ELISA, and culture. And the positivity is only about 2%. And in animal sample, it is about 16%. We had about 87 samples positive out of 643. And if you see the serovar prevalence, in humans, we see Australis and autumnalis, followed by ectohemorrhagica and javanica. In animals, we have Australis and autumnalis again, followed by canicola, javanica, and cryptophytosa. So this is the serovar thing. So moving on to diagnosis, we started, we had a project with DST and we wanted to develop a diagnostic kit. And for that, our approach was to use a multi-epitope protein. So we chose the epitopes from 4 different proteins, LipL41, LipL21, OMP-L1, and we took the epitopes and we synthesized a gene with the linkers and it had 8 epitopes from 4 different proteins, and it's a 480 base pair sequence that we cloned and expressed. And we also did blotting to confirm. And we developed an ELISA using this multi-epitope protein. And the sensitivities and specificities were quite good. It was about 93.3% sensitivity and 96.6% specificity in comparison with the gold standard, the MAT test. And this antigen or this kit was transferred to genomics laboratory, and they started using this kit in the form of in the form of lateral flow test. And the feedback that we hear from them is, even though this protein was working well, they tended to mix this multi-epitope protein along with the LPS extracted from the individual serovars, and then only the lateral flow was working better. So as Dr. Manjula said, LPS was the most immunogenic protein. The more immunogenic fraction, and using trying to replace individual proteins in the diagnostic format doesn't work all that well. That is even though we used epitopes from 4 different proteins, again we needed LPS also to be added to work better in the clinical settings. And we also showed that this multi-epitope protein could pick up the antibodies against different serovars, which we identified by MAT. It was almost like 94 to 100% reactivity could be seen to antibodies to different serovars. And also another feature that we could come across is, see, the commercial kit PanBio is an IgM ELISA kit. And we found that in endemic places like Chennai, the IgM may not perform very well because already you have a background of IgG. So, it's very difficult to detect IgM unless you use a class capture format. In an indirect ELISA format, even though you use anti-human IgM conjugates, the IgG will, due to the interclass interaction, will really reduce the IgM reactivity or the color. So we have seen that in many cases, MAT positives had more IgG rather than IgM. Although we know MAT is agglutination test, IgM is a better agglutinating antibody because of its pentavalent structure as compared to IgG. But in reality, in the background of IgG, we could not really detect a lot of IgM. And the kind of application of this IgM ELISA in an endemic setting is really questionable. We found more correlation with MAT with our IgG ELISA rather than IgM. And we also did some meta-analysis with various huge publications in terms of antigen assay formats and so on. And what we found was the best. We have compared different assay format, different antigens, IgG, IgM ELISA, single and multiple assays, and the combination of antigens that we did. So you could see the latex had the highest sensitivity, pooled sensitivity. Obviously, probably it is detecting IgM better, followed by the flow through as well as the ELISA. And in terms of antigen, we had this R-GROW-EL to have the maximum sensitivities, and both IgG, IgM performing equally well. And multi-antigen assays was much better than single antigen assays. And in that multi-antigen assays, we find that the highest sensitivity was seen between R-OMP-L1 and R-LIG-B, as well as highest specificity also using this combination. And we have used the kit to just do zero surveillance among the risk group people. So we have taken people working in various slaughterhouses in Chennai, 115 samples, and we found that there was about 38 samples, like 33% positivity among this high risk group people. And we also did a kind of survey in various labs that offer leptospira testing in different places in Chennai. And we did a survey initially to know what are the kits they are using. And we found predominantly they use 3 IgM ELISA kits, PanBio, Vidion, Cidion, and Sky Medex, and 3 different lateral flow kits from 3 different companies, J Mitra, SD Card, and CTK. And you can see the kind of usage pattern and the cost also. Just because it's a lateral flow kit, they don't reduce the cost. The cost is almost as similar to that of ELISA. And this graph Venn diagram shows you that there are 33% using IgM ELISA, 45% use rapid test, and 21% use both the assays. And what we did, yeah, these are the kits. As I said, we used 6 kits, and what we did was we took 50 samples, of which 30 were positive and 20 were negative in MAT. And then we used these 30 samples across all the 5 tests, 2 ELISAs and 3 lateral flow kits. And we found strangely that only 21 samples were positive, which were 30 out of 30, which were positive in MAT. Only 21 were positive in PanBio, 18 in Cidion, Virion, 21 in SD Card, 26 in J Mitra, 25 in CTK. And now with our multi-epitope, there were more positivity. And negative samples also had different variations. So when we started analyzing the kind of, these are the data that is shown here, positive and negative, using the 2x2 contingency table. And what we see here is the claimed specificity of these samples of these tests are so high. But what we have observed, sensitivity with MAT is very, very low. And we have also analyzed that in the absence of gold standard. So without taking MAT as a gold standard also, the sensitivity and specificities are much, much lower than what has been claimed. Probably most of these kits are imported, and these are probably validated in a different target population where Leptospira may not be as endemic as you see in our country. So this is our results. And if you see, even the correlation between tests was very, very low. Only PanBio and Cidion both are ELISAs, they correlated only about 63%. Whereas, of course, correlating ELISA with lateral flow may not be a good idea, like correlating apples with oranges. But still, in terms of the positivity, you are using either of these tests to assign a clinical sample as positive or negative. So in that context, when we correlate the result, the percentage of correlation was very, very low, as is shown here. And this slide is very important. When you see here that only 3% of that sample, which means out of 30, only 1 sample that was MAT positive was positive across all the 5 test formats. And that is only 3%, and 60% of the samples which were negative were negative across all 5 test formats. So this is the kind of assay that tells you the wide variation that exists among the clinical, among the tests that are diagnostic kits that are used for testing clinical samples. And obviously this will be reflected in the test results on the clinical samples also. And 40% of the MAT positive samples were not able to be detected in any of the 5 tests. So this is our result, which shows that the situation is quite grave in terms of application of these diagnostic assays and kits and platforms for clinical diagnosis. And with this background, I should also mention here that we also compared MAT across 2 different institutions that offer MAT in Chennai. One is ours, one is another medical institution. And we sent 25 samples which was tested with MAT, and 7 were positive in our institution, whereas in the other institution only 1 was positive. and there was a wide difference in the way the MAT is being done. This I need to highlight. Although MAT is considered and told as a gold standard test, the way the MAT is performed probably is not synchronized in a concordant way, because we do that with 12 zerovars. The medical institution does that with only 1 zerovar. We do it at different dilution. They do it only at a single dilution of 1:100. So there was a wide variation, as we see in diagnostic kits also, wide variation in the MAT results between two different labs. We had 7 positive out of 25. They gave only 1 positive out of 25. And it was positive to different zerovars here. They don't do zerovars, so we don't know the zerovar specificity of that MAT result. So in this backdrop, I've just highlighted some of the challenges and solutions. There are several commercial kits available, as I showed you, and MAT is only done as a selected lab. But what is lacking and what is needed for the Indian scenario is there is no competence testing. Nobody knows what is the quality of MAT that is being performed. There is no uniformity in the zerovars that are used, no SOPs that are followed across different labs, and nobody tests the purity of the strains. And the availability of reference strains also is a big question. If you have to import, it's quite expensive. And these have to be inbuilt into the diagnostic lab. So there has to be a consortium of labs that has to be created, and there should be free exchange of materials, both strains as well as sera samples. There should be interlaboratory comparison as is warranted in NABL accreditation. And also we should try to slowly replace the zerovars, which have been standard ones used for many, many years, with some local strain, because there are publications which suggest that use of local strains are much better in diagnosis. So the need of the hour is to form a consortium of diagnostic labs and do a competence testing and follow uniform procedures in MAT. Only then you could say that I am using MAT in a gold standard way. And coming to vaccines, as is known, in India, there are only canine vaccines and only one company makes, that is Indian Immunologicals. They also use only the reference strain. They don't use the local isolates. And bulk producing this vaccine in a kind of fermenters and so on is a big challenge, because the organism is motile. And developing appropriate model, although hamster models are being used, but we don't have a kind of a approved system for vaccine approval. So we need to concentrate on these areas, try to apply bulk production strategies, include isolation of local strains, as Dr. Syed Faisal also was mentioning. But in that context, again, I would like to share a small knowledge or learning. We worked with the National Institute of Epidemiology, and they say that they have lots and lots of strains that are isolated from humans. So when we said that we are not able to isolate from animals, so their approach was totally different and unique. So what they said was they had actually taken certain villages into their context, and any fever that comes to a person in that village is reported to that institution, and immediately they go and collect the sample. But what we do is, see, there's less linkage between our hospital in the veterinary colleges and then the clinical lab. And the people who bring the patients, to the hospital are being treated as any other fever or pyrexia of unknown origin, given antibiotics. If that doesn't subside after 4-5 days, only then they look at leptospira, by which time, the leptospiremia is gone and you are unable to isolate the organisms from the clinical sample. Of course, we have done a lot of efforts and we have succeeded in isolating one canine sample, which we have full genome sequence also and submitted. So this is a big area, the kind of sampling strategy that needs to be followed. This can only work if there's a very good linkage between the clinical lab and the laboratory. And in human cases, we hear that they are able to easily isolate because they target very early the sampling before any antibiotic is given at the peak of temperature. And of course, the other areas is non-availability of local strain. So if this is really overcome the sampling strategy, then it may be possible to isolate more and more local strains. And using a local strain in a vaccine would be a better method of come up with a different good vaccine with the locally isolated strain with a different developed bulk culture conditions. I think these are the need for vaccines and diagnosis and isolation. And this is what we have learned from our little experience. And some companies recently, we also validated a LAMP assay for a company called Agape, and using the DNA that we have from and also the serum samples we had validated, and that seems to be working. It's based on a isothermal amplification LAMP assay, and we are concentrating our efforts into isolating local strains of Leptospira by establishing a more closer link between the laboratory as well as the clinics. Thank you, sir. Thank you for the opportunity.
So now I welcome Sai Kumar, sir. So please give her something. Sir.
Okay, thank you. Yeah. Let me try to share my slide here. Now, can everyone see my slides in full presentation mode?
No, sir, it is not in presentation mode.
Is it not in presentation mode?
Please share entire screen, sir, while sharing.
Yeah, share screen. Now is it all right?
No.
Yes, I think so.
No, not there, sir. Again, unshare the screen.
Stop share.
Now your presentation will be there. Start slideshow in your presentation. After starting slideshow, now share the screen by pressing start button. Share the screen and then select the slideshow mode option, sir. In your system, start slideshow. After starting slideshow, then share the screen.
Yeah, just give me a minute.
Yes, sir. Yes, sir.
Yeah, now all right?
Yes, sir. All right, sir.
Yeah.
Yes, sir.
Sorry for the inconvenience. Good afternoon, everyone. Yeah, most honorable chief guest and our esteemed senior Professor K.M. Gujarbarua sir. So also today we have amongst us our director as guest of honor, Dr. Triveni Dutt sir. Very distinguished director of NIIB Hyderabad, and esteemed scientists from different institutes, those present here, Dr. Dinakar Raj, Dr. Vijay Chari, Dr. Siraj Ahmad Khan, Dr. Manjula Sitaraman, everyone on board here as participants and experts. At the very outset, I thank everyone from amongst the organizers of the NIIB for inviting me and giving this occasion to share my views on this also very important zoonotic disease in this country, and to apprise the audience about the current status and especially the challenges that we meet here. I'm unable to move my slide. Okay. So India is a very fertile country with about 1.4 billion population now as on this date, and the challenges that it throws open to us are on very many fronts. It's really troublesome to govern and take their interest of this burgeoning population. And the predictions for 2030 or 2050 are also bearing on us all the more to make them feel healthy or to keep them healthy by providing the much needed nourishment that is required to keep this population healthy. There have been many issues that we talked since morning with the opening discussion held by Professor Hegde there. So also our honorable chief guest also did allude to the challenges in this country in terms of the emerging zoonotic diseases and how serious a threat they pose. And from amongst the newly emerging diseases and some going on to become pandemics, about 75% of them are of zoonotic origin and all that. For the first few slides, next few slides are those pertinent, the drivers that drive this kind of disease that we are talking about will only be referred to in my presentation. So, as we struggle to keep and meet the challenge of food security in this country, more and more of this forest land is also being claimed for agricultural purposes. And as a consequence, there is some climatic change, adverse impact on the climate. That also acts as a driver for some of these vector-borne diseases, especially if we are talking today. It is said that as we go on to multiply at the rate that we are doing, the challenge to feed them, this burgeoning population, would be all the more on the agriculture sector. So our population would demand that we increase our food production by 100%, be it for food grains or from livestock products that are essentially required for proper nourishment of this population. Very quickly and briefly, I would like to reflect in this context that we have, through our compulsions to meet these food requirements, we have been changing our agricultural practices. Plus, also there are other factors which dictate that what a farmer should go for. And here is this typical situation that if we talk of the changing trend in the farmers of Punjab state, that more and more are taking to paddy cultivation, which was not the traditional crop there. Because basically the population resident in Punjab and much of North India take to consumption of wheat, not rice. But the economic compulsions and the remunerative price they get drives them to more and more these basmati rice production. As a consequence also, this is as a direct result in the bottom paragraph, I have just given one reference here, but there are other episodes also being published that GE is also being recorded and in the sentinel pigs also to what extent whenever studies were taken up. In a sense, where this disease was not found is finding new ground because of the anthropogenic drivers that are making it conducive for the vectors and the multiplication of the viruses in them and its transmission to humans. Now we know much of our rural population, 65% of this country's population reside in the rural side, countryside, and it is there that this disease and diseases like these have the maximum impact. So public health authorities have time and again emphasized on these, and I also reflect on this very briefly that there's too much that favors these kind of organisms and the vectors that drive these diseases of genotypic origin and transmitting them to even the elite in the urban sectors. The transmission cycle has been known for quite a long time, and we know how it circulates. But there is a new dimension which was not so well known, is now being known through extensive studies. It is that the bats are also responsible as a reservoir and for occasional transmitting or giving us a new variant of the virus or one that is strictly restricted to these species are now finding way to the humans and animals. Such like studies have been very few in this country, but there is enough evidence that bats in this country are also harboring these viruses because serological evidence is there, and that kind of information goes back to several decades ago also. I must add here that we also tried from our laboratory at IVRI to investigate these bat specimens for presence of flavivirus and especially JE viruses, but we did not succeed. Most likely, I think it is because of the small sample size that we examined. But it is one area what I want to highlight, that there is so much that has not been studied in this country. The disease first reported from Japan in 1871 and in India in 1955, meaning it's several decades ago, more than half a century ago that we have come to know about this disease. And time and again, studies intermittently done or on a large scale in recent decades summarize that this is a disease that is endemic in this country. It was restricted to south of India for several decades until it jumped to other parts in the eastern and northeastern region in the 1970s. And for quite some time, Uttar Pradesh was thought to be the most endemic and hyper-endemic region, especially the Gorakhpur and other regions where massive outbreaks have been recorded time and again. But now we know the picture is quite different and it is expanding, and now we call it as an emerging zoonotic disease in this country. There is this kind of opinion that the cases in total number only a few thousands, but the impact it makes sometimes shakes up this country very badly. And I mean, the impact on the socio-political fabric of this country is immense. So also that thousands of lives are lost, if not thousands, several hundreds. But what it should be recognized that if we record 1,000 deaths, equal number of sequelae, neurological sequelae, it leaves in the children, leaves them incapacitated for the rest of their lives, so which is a heavy burden again on us. Now coming to some of the challenges that I want to draw your attention to. Look at this slide here. It is from the Medical College in Gorakhpur, named after Baba Raghav Das. 2011 and 12, this chart depicts when it peaks. And look here that the positive samples in dark black were first noticed in June, and for the rest of the samples during the peak and until it subsided, there wasn't any confirmed case of JE there. Most cases are now here, as in this figure, published figure. It speaks about acute encephalitis syndrome. So numbers are very few that could be confirmed as Japanese encephalitis, AES and Japanese encephalitis. This is a book that recently got published from a doctor named Dr. Kafil Khan, who worked in this Gorakhpur hospital. A book that worth reading to grasp the immensity of this problem, the tragedy it unfolds whenever it strikes, and it does strike year after year, and for decades it has been going on. It's a good book for those of us who are engaged in this research. Dr. Kafil Khan was working in this ward that is named as AES-JE ward. 100 number ward is because it has 100 beds there for children. And every year, or for the years, this is a report for 2021 in October that says that encephalitis cases have declined in Gorakhpur, yet it still cause for concern. Out of 291 cases of AES, 30 cases were only confirmed to be Japanese encephalitis. So what is this AES? Acute encephalitis syndrome. Clearly, it is not a disease. It is something that is causing death and all this catastrophic public health impact from etiological agent which we purely understand. That's why it continues to be labeled as AES. AES in Japan, and now there were some previous reports by our director also, and so also mentioned by others. Assam accounts for 60% and 76% of the AES and JE deaths in the country. This is a shift, a recent shift that has happened. It was there in 2014, and in 2021 also, we have come to recognize that it is a burning problem there. AES, of all things, are a very big number. JE, a small fragment of it. It was only recognized as a notified disease in 2014. In Bihar also, it is known by many names, and year after year, it's a serious disease that causes a lot of embarrassment. All kinds of investigations have been carried out, and it's still not clarified that to what proportion it is JE and or what else it is, but we continue to label them as AES. But what? There are cases of similar AES kind that has been reported here. Look at one. This is not the only one, but from Kerala, caused by West Nile virus. West Nile virus is another flavivirus detected and confirmed regarding its presence by the ICMR Institute several decades ago in this country. And there are so many cases of AES, and very few of them have gone into detail to study, rule out, confirm, if not JE, is it West Nile? And how many of them do turn out to be West Nile in Uttar Pradesh or West Bengal or in Assam? In West Bengal also, there is this report of West Nile virus from amongst JE cases, suspected cases. There are other flavivirus also rampant in this country. We know we are not new to dengue. So how many of these, and with the methods that we are applying for diagnosis and confirmation of these AES-like cases, what fraction of it is contributed by West Nile virus? Some attribute to dengue also, but how many of them? Are our diagnostic methods appropriate to take into account all the possible agents, be it flaviviruses or other agents also that are likely to contribute to these AES cases? So that's a challenge. Now we know from this country, and this has been discussed, there are 5 genotypes known worldwide, especially the G3 and G1 viruses, that genotypes 1 and 3 that we have been talking about, and that including the scenario that is changing in much of other Asian countries. Here also G1 has come to be recognized in UP and West Bengal, and as mentioned earlier in 2009 and 10. So the changing trend of this virus, though this is not a virus that changes so very frequently, going by, because it's after all a flavivirus, but quite a stable virus, though it's RNA virus. RNA viruses are likely to change, mutate as frequently as possible. This map is a picture showing where all its presence has been recorded. But strange but true, we know for all these years that pig is a big culprit, amplifying host, has been a target sentinel animal for monitoring, and so much has been studied for all these years from the medical side, from the veterinary side. But in 2016, our laboratory for the first time reports that here is this disease among pigs, and what is written in the standard textbooks of veterinary medicine is recorded here amongst pigs, that it is associated with reproductive failure and neurological disease in unborn piglets, fetuses in utero died, stillborn, delivered stillborn, mummified fetuses. All this happens in 2016 for the first time. Why I emphasize, why I say this, besides telling other things, is that how little we cared for this disease from the veterinary side is what I want to emphasize. Despite knowing full well that this is primarily driven by mosquitoes and pigs. Human is only a dead-end host. And when we did make an extensive study after complete genome sequencing and all, we related to all that we have known about JV circulating in this country, detected in humans, mosquitoes, and pigs. Pigs, there wasn't any information available. There was only one report of a virus detected in pigs, but nothing known about its genotype or any sequence for comparison today. Already emphasized, highlighted by Dr. Himani earlier in her presentation. So all this led to the conclusion that yes, it is the very same virus detected several decades ago in the 1960s in Japan in pigs, continues to be circulating amongst pigs in this country, no different. And it is very closely related to the human epidemics, to the virus circulating and reported by ICMR Institute from mosquitoes and humans. Our diagnostic options today stand on mainly ELISA-based methods for detection of IgG antibodies or IgM antibodies for detecting recent infection in the face of an outbreak. And both these antibody methods have also been developed at this institute from our Veterinary Public Health Institute. And you have heard Dr. Himani speaking as a collaborator in this our presentation today in the DBT project. It is from her laboratory that these two methods have also come and are available for commercialization and application in the field as well. Our laboratory and also from the public health laboratory, TACMAN assays for much better sensitive detection of this agent, because there seems to be a gap we doubt that from among the so many AES cases, why only a small fraction of JE cases are being detected? Is it because of the low sensitivity of the diagnostic test, hence the requirement for this kind of test? And we clearly now have shown with the data also, Himani was speaking today, that it is much more sensitive and can detect even one or a few copies of this virus. So better we adopt this kind of technology for diagnostic application in humans. This was not discussed today in this presentation. Maybe it wasn't a part of this project, but I gathered that NIB has also developed a biosensor based method for diagnostic application, which would detect basically the non-structural protein, which is expected in new recent clinical cases for diagnostic application. I think there is a need for putting it in real hospital-based application so that we know how well it performs, and hence there we can do necessary refinement in the test before it can be recommended for clinical application in the hospital setting. My opinion here, there are so many dimensions in the project that we were working through this DBT project. The emphasis one was on diagnosis or for finding therapeutic molecules for treating these cases. But in my study, whatever I did, and in this presentation, I would like to emphasize on JE control through vaccines. This has a serious public health impact and all of that we know, and that this vaccine does give an option to prevent it in the first place. And there are several kinds of vaccines, all kinds of vaccines that we can think of or today technologically possible that have also been used to develop vaccines. So they are listed broadly here under the 4 heads seen in this slide, and we shall discuss very, very briefly one after another. The earliest prototype vaccines and were used for several decades was the formalin inactivated mouse brain vaccine, which was produced in all the Asian countries and so also in our country. And it was only stopped in 2007. Until then, starting from 1955, when the disease was recognized, and some decades later, we came up with this vaccine, the mouse brain vaccine. And it was discontinued for reasons that we know full well. I don't have to elaborate on that, but it certainly was a good bargain despite the drawbacks it had to control JE among children. Now, later when we were able to develop the cell culture based systems to grow JEV, then came these next generation vaccines, cell culture based vaccines, but largely inactivated vaccines. So here I have tried to list all those important manufacturers and the products that were used across the globe, including in countries where it is not a problem, but still used and recommended, for example, in much of US and all, because there are travelers who go to these places and they are recommended to take this vaccine before they put step on these endemic JE endemic countries. And of all things, if we talk that it is the SA1414-2 vaccine that has been in use and recommended worldwide. It's a vaccine strain that was neuroattenuated through different passages in the cell culture. And originally it was from mosquitoes that it was isolated, where parent wild type virus. And subsequently it came to be in use across the globe, including in our country. Inactivated SA1414-2 virus-based vaccine is produced in our country by Biological E at Hyderabad. And GenVac is the first entirely indigenous vaccine that Bharat Biotech is producing and making available in this country for the children. And this is from the NIV strain that was isolated several decades ago from human subjects. One quick word, I would not like to go into detail. This vaccine is the most widely used vaccine in several countries, as can be seen here in the first paragraph. And this company makes close to 30 million euros from the sale of this vaccine, Valneva. The figure is from 2015. This is what we have also come to be producing in this country, the SA1442 based vaccine from Bharat Biotech. Sorry, from the biological E. Okay. This is the Genvac from Bharat Biotech, what I referred to. There are live attenuated JE vaccines also derived from the SA1442. And this is also now basically a Chinese vaccine that is now available commercially for any takers. And this country has also imported from China for use in the immunization, national immunization program in this country. A single dose vaccine also favored and licensed in Nepal, Korea, and Sri Lanka. Research is also underway to develop genotype 1 based vaccines, and there are some which are still in experimental stage. The reasons have been cited by previous speakers. I'll skip this. This is to say that when this vaccine was used, how it could help bring down the incidence of JE in humans. There are other experimental vaccines, and this is a good one, claimed to be licensed already in Australia to 2012, developed by Sanofi Pasteur, a recombinant vaccine based on the yellow fever virus backbone with the SA1414 pre-RM membrane and envelope E gene combined with it. A good vaccine. Said to be much better than the SA1414 vaccine. Details not required here. You can find it out. There are other vaccines on all other platforms that one can think of and have been used as a research tool for developing new generation vaccines, and they have all been tried to develop new generation JE vaccines. Much of this effort did not result in a very good vaccine on this vector-based vaccine on Vaccinia platforms. So let's not spend time on it. So also the recombinant protein vaccines that have been worked here in this country. Also the last one about from NIAV, Sudhanshu Vrati and his team. These have not been translated into viable products for use. Virus-like proteins also, some research has been done, but nothing yet as a product there. Plasmid DNA vaccines reports look promising, but not a product that's already translated for commercial availability. This again is another on the research front only, and it doesn't translate into a product there. So to summarize, in India, to deal with this endemic disease, a preventable public health disease, a vaccine preventable disease, we have only very recently come to be producing these indigenous vaccine, the Genvac, not in adequate number to be self-reliant. There is another one based on the SA1442 by biological E. But yet we had to be, sorry for the word, begging. Yeah, that is how the reports have come to, if you read it in any great detail, to meet with the challenge that is unfolding year after year, and to drive our vaccination program and our commitment to deal with this vaccine. Sometimes we were late and China told we are already booked. You should have booked earlier. We can't make it available. Such was the embarrassment that this country still faces. Now talk about vaccines for animal use. I would like to spend the next few minutes on this. There's this JE vaccine based on the Anyang 300 strain in Korea, used for more than 3 decades. There's also this new live attenuated JE vaccine from a G1 isolate adapted to vero cells, developed again by now in Korea. Another strain also has been used to develop inactivated vaccine for use in pigs in Korea, reported in 2014. There's this inactivated JE vaccine containing a recombinant protein to further boost the response. And this has been again intended for use in pigs. In Taiwan, Japan, and Hong Kong, a live attenuated strain has been used to prevent reproductive failure in swine. A study conducted in Bangladesh, of all places, in Bangladesh, in our neighborhood, with the help of experts available worldwide, including those in America. This combined study shows the importance of potential impact of pig vaccination in reducing the disease burden in the environment. The brief of this research says that when it was estimated, they found that JV infects 20% of the pigs, and that the basic reproductive number is about 1.2. And as per the model that they tested, when 50% of the pigs are vaccinated, it would result in estimated 82% reduction in annual incidence in pigs. Such like study has not been conducted in our country, where JE is a much bigger problem than in Bangladesh. But still, what I want to emphasize that there are experts who think and who have used this vaccine in pigs to reduce the burden of disease in humans. So it is because of my primary concern and interest in these zoonotic diseases. And I used to be greatly disturbed with year after year and these AEs cases, and how so very few cases are amongst them are JE. And then looking into this aspect, I did study in pigs, considered all kinds of agents that can be transmitted from pigs. If not JE, there could be other agents, including enteroviruses that are attributed as a causative agent for encephalitis in humans. So I studied enteroviruses also in pigs. So I'll leave that aside, but talk about JE, then I and our students here, we succeeded in isolating the JE virus, and I proposed to others at this institute to take up this vaccine work, because now it is available, the virus is available for the first time in the country of a pig origin. So we can make it. But the responsibility finally came upon me. Otherwise, I'm not a vaccine guy, I'm a disease specialist. But whatever, we went on to develop a killed vaccine. Attenuated vaccine will take much more time, but killed vaccine has been used in humans and pigs for such a long time. So that is a quick, easy method. We did take up this study in mice, and including challenge studies, and all immunological parameters were studied. And we found with the combination of adjuvant and the dose rate, etc., that I wouldn't like to go in much greater detail. It seemed very promising. That led to further studies in pigs and assessing the humoral and cell-mediated responses in pigs, which are also quite promising. And we now have a vaccine candidate which can induce an immune response that is robust and we studied that for only 60 days. But during that window period, we found that it does induce a good neutralizing antibody response in pigs. Only some to support these findings, that the virus grows in cell culture in viro cells to a very good titer, appreciable titer, expected titer, up to 7. And that when we did these mouse experiments, it induced a very good IgG response in be it in antigen alone or when given with the montanide antigen.
Sir, sorry to interrupt you. So we are running late.
Okay. So I'm also coming to an end. That this vaccine, when tested in pigs under the conditions that are reported here and all parameters that we studied, we found it induces a good neutralizing antibody response. And all other parameters were also indicating that the CMI response is also robust. So I'm not going to hold the audience any further. That is disease that's quite embarrassing after all these years and several decades that we have put up with. We should be doing something else. And what is that? Think differently this time. Develop the necessary vaccines that can be used in pigs to reduce the environmental burden and therefore the disease in humans also. Either way also, it would also contribute to reducing the disease in pigs and horses also. They are dead-end horses and they meet with a fatal course, sure for death, and they also are very precious animals. So we should have a vaccine for animals. Research should be oriented towards developing a good attenuated vaccine. I already depicted that how a good vaccine like SA1414 makes several million euros in the world market. And if we are capable, we should also devote time towards developing a good attenuated vaccine, which will find not only market in this country, but in the rest of the world. Thank you for your patient hearing. Thank you.
Thank you. Thank you, sir. Thank you very much. So now I think we are already very late. So there are two more talks, Dr. Vijayachari and Dr. Siddharth. So we will continue this talk after lunch. So we'll break over here and the next session will be coming session, which will be the boardroom. So we will get back at 2:30 in the boardroom.
2:30 now.
2:30. I think we can just delay a little bit. 2:45 or 2:40?
I think 2:45.
2:45. 2:45 we will be in this session. Only experts who are online and we came over here, and the project investigators are part of this brainstorming session.
Do you like to thank us?
Yeah, thank you so much. I want to thank everyone who has joined online, all the students, fellows, and experts for giving their time. And thank you so much. Thank you. I would like to thank Vibhava sir for giving his precious time and agreeing to our consent for coming over here. Long way, sir. Thank you so much, and I'm very much thankful for giving the feedback at the expert comments, which will be very useful for us to move forward with our research. So nice of you giving this time and being part of this workshop. Certainly, your presence over here made this workshop successful. So I will again thank our director also. Thank you so much, sir. Thank you very much.
Thank you, one and all. I take leave now.
या फिर बंद कर दो।